think Novashin is the guy cytologically. But what we showed is at the whole chromosome level, that
Yes.
based on the chromosomal location, these 400 copies of genes are turned off, and on the other chromosome they're turned on. That kind of mechanism we showed it in Arabidopsis. So those kind of studies.
How does this silencing occur actually?
How does it occur is we have shown that it is those regions are hypermethylated. So that is a condensed chromatin is happening for sure. But the very intriguing question is why it is although sequences are so similar, why one chromosome is chosen
Yes.
for silencing, not the other.
That's right.
Yeah, and in at least Columbia's ecotype, we are hypothesizing that there is a 75 kilobase of transposon region next to chromosome 2 NOR2. Okay, but in chromosome 4 there is nothing next to NOR4. That is one hypothesis we have. Is it possible that silencing from the adjacent transposons is spreading to the NOR2, and that is what is causing its silencing? So for that you have to really clip it off of the chromosome, which is a very precise chromosomal engineering. Picard lab has been trying for the last few years. I think that is something they want to really test. Is that how the silencing is spreading? But other than that, some epigenetic modifications are involved silencing. But the interesting question still remains to be answered: how it chooses one chromosome over the other when the sequences are so identical?
Any other question for him?
Yeah, sir, I have a quick question, sir, if I'm allowed to. Yeah.
Okay.
Yeah.
Please.
So Girish, I just have a quick comment. When you said ONT, that was a kind of an interesting thing which you were also mentioning earlier. Could there be a correlation seen with single cell genomic data, Girish? Because I was just checking some papers over here. There are wonderful articles that have really come up, taking the single cell genomic, capture-based methods, the sidelines of the ONT that we have discussed. So it's been some time, yeah. Would that really bring out, a kind of a reason and understanding whether or not, silencing is seen only in certain, genes?
I think, Prash, it's an interesting question because there was recently one review I was supposed to review. I think it's getting under publication. What they are shown is there is a tissue specific expression of some of the ribosomal RNA genes, which people have not variants particularly. Now, if you want to show that precisely in any other organism, I think a single cell based genomics would be extremely helpful in answering those questions more precisely. Right now, at least there has been a first report being they're trying to publish. It means that it's not published. At least I heard that they got this data to show that there is a. So far, we were not having any data. Of course, we did not work in that direction necessarily with extensive analysis of genomes because but
Girish.
it is sir.
Okay, I will hand over to Vijay Gehlot now.
Yeah.
Invite the next speaker.
Yeah.
Vijay?
Sure.
Yes, sir. Yes, sir. I'm there.
So, thank you very much.
Thank you, Grace.
Thank you.
Okay. So, a very good afternoon to all. So, in this session, our next speaker is Professor Rajiv Bandyopadhyay. Dr. Bandyopadhyay currently is a professor in Department of Botany, the University of Burdwan, India. He is BSc and MSc in Botany from Burdwan University and North Bengal University. And he also did PSc in Botany from University of Calcutta. He has more than 17 years of teaching and experience with postdoctoral experience in CCS University, Meerut, CBN University, South Korea, and University of Georgia, USA. He has several national and international research awards and recognitions, including prestigious Bioclues Fellowship in 2007-8, South Ocean Expedition 2011 by Ministry of Earth Science, Government of India. He is also a member of 10 scientific body and editorial board member of several international laboratory journals. He currently involved in the research on extremophilic microbiology, algal biotechnology, molecular markers, and plant microbe interaction, plant pigments. So, and today's topic of his talk is bioinformatics, a novel way to study microbial lifestyle. So, Rajiv sir, please.
Yeah, I am here. Thank you for the nice introduction. But can Girish close his presentation so that I can show my presentation.
Girish, did you close? No. I think he's
Hello?
unmuted himself.
Hello, sir.
Yes,
Can
Girish,
you,
did you close
Oh,
your
yeah, yeah, sure. Yeah, sure, sir.
so I want to thank Prashant and Bioclues member. Let me present. Slide presentation. How to do it?
Rajiv, make it full screen.
Yes, sir.
Yes, fine.
Is it okay? Is it okay, sir?
Yes, fine. Go ahead.
Let's see. So, thank you everybody, and I am humble to Professor T.K. Gupta, who is chairing this session. So, bioinformatics, my topic is bioinformatics, a novel way to study microbial lifestyle. So, for the beginner, I am making these slides, first two slides, because bioinformatics, a lot of people, they have no the clear idea. They just know the wet lab. So bioinformatics, it's a computational tool we use for the molecular data, and biologists normally they used to develop or generate the data in the laboratory in the form of DNA, protein sequence, genes expression, etc. And computer scientists, they are expert to develop different tools, softwares, algorithm, and store the data in different databases. The bioinformatician normally they bridge the gap between the biologist and the computer science. They know the biological phenomenon. They know to make the question or make the problem and how to answer with the help of the wet lab data or interpretation of the molecular data. So normally, for a simple person or layman, bioinformatics, they have major function like searching of biological databases and compare sequences. It may be DNA sequences or the protein sequences, and they can estimate the molecular structure, different protein structures. So this is a very simple idea about the bioinformatics to a layman. So I am telling here a different story. That is my title, The Lifestyle of the Microbes. So, bioinformatics is a weapon what we used in our study to know the lifestyle of the microbes, because microbes they may be anywhere and their lifestyle may be different. It's not like others. Say, for example, bacillus species. So there are different type of bacillus species, but all bacillus are not same, and their lifestyle are not same. So we can study in the wet lab, but as well as the bioinformatics weapon can help us to understand in a better way for their lifestyle. So I can remember when I used to work with Professor Gupta at CCSU Meerut, he used to advise us, design your experiment like a way there may be component of wet lab and bioinformatics, say 70-30, say 60-40, or say 50-50 like this. So long back in 2003 and 2004, we got this kind of training from Professor Gupta, and till we are carrying the concept, and we are really trying to making more bioinformatics in our work. So mostly I worked in plant system most of the times, but for last 6 years I am working on microbiology, and mostly the extreme microbes. So here I have chosen two stories of the microbes and their lifestyle. Say for example, the first one or first case is a Bacillus species. We identified this thing as a Bacillus altitudinus. It was reported from Southern Ocean during my expedition in 2011. So earlier, many scientists like Sivaji from CCMB and scientists from ISRO, they reported Bacillus altitudinus from stratosphere. So in the present slide, you can see the different strata of the air, that is stratosphere, troposphere, and the sea. So they reported the samples from the air, but I reported the samples from the deep sea of Southern Ocean. So when we participated in the Southern Ocean Antarctica expedition, so a lot of scientists from abroad and a lot of scientists from India. Earlier, they also reported many bacterial species. Sivaji itself reported 28 new species from Antarctica land. So we have mapped them in the recent map, and from the Southern Ocean area, from this area, we reported some new bacterial species from Southern Ocean of different depth and different locations. So according to the station, different stations, we have numbered the stations, station 22, 23, for example, 25 and 26. And we mapped all the isolated bacteria in a particular stations, and left side is the different depth of the ocean, and Southern Ocean is almost 4,000 meter depth. And we reported one bacteria, that is Bacillus altitudinus, from 3500 meter. So this is the sampling area, or the detailing of the station, their temperature, their wind speed, and different things. Morphology, biochemical study, and different instrumental study are there. I am not going in details with all those kind of studies. I am focusing only on the bioinformatics tools. So anyhow, this sample is very interesting, because these species, for two reasons, it is important and interesting. Number one reason is that Sivaji and group from CCMB and scientists from ISRO, they reported this kind of bacteria from upper area, that is from stratosphere. But surprisingly, we got this kind of bacteria at the deep sea. So how is it possible from a long range or variation of their habitat? And number two, while handling these samples in our laboratory, we found that this is highly UV resistant. So, for example, our general sample in our laboratory, what we have collected from Imtech Chandigarh on the NCCS Pune, that is different bacillus species, they can resist UV up to say 12 minutes, 15 minutes, or 20 minutes. But this sample, it can resist up to 90 minutes or even 120 minutes under UV. So this kind of peculiar thing, or this kind of some absurd thing haunted us, how to give the reason and what may be the reason. Then we took the help of bioinformatics. So these are the regular practice in a microbiology lab for the wet lab analysis. I'm not going in details, just I am showing the picture to understand the bacterial size and shape and their surface. So all these are morphological characters under different microscopy, that is fluorescence microscopy, atomic force microscopy, and transmission electron microscopy. So it looks like this. So I'm not going in detail with the wet lab experiment or other data. I'm just focusing the story, how bioinformatics help us to understand these sample and how they behave in a different way. So we went for the whole genome sequencing of this bacillus, and we named this sample as Bacillus altitudinus SORB11, that is Southern Ocean expedition in 2011. So we isolated this sample from Indian sector of Southern Ocean, and approximate genome size is 3.6 MB, and the 16S rRNA gene length is 1500 base pair when we analyze the whole genome data. But when we extracted 16S rRNA gene, the size is 1461 base pair long, approx 1500 base pair, and it showed similarity with Bacillus altitudinus, Bacillus stratosphericis, and Bacillus aerophilus. All are reported from the upper surface of the air. But our sample is from deep sea, and GC content is approx 41.2%. And interestingly, these samples also produce a good quantity of exopolysaccharides. So then we went for the brick analysis, that is blast stream image generator analysis with the help of different genome of Bacillus altitudinus and other Bacillus one. So they showed the similarities and the dissimilarities, and we can compare it with the other genome. And in this picture, the black box regions are there. We mark them as 1, 2, 3, 4, 5, 6, 7, 8 like this. Now, where the variation lies? We compare our Bacillus altitudinus with SORB11 with Bacillus altitudinus 41 KF2B, Bacillus stratosphericis, Bacillus stratosphericis M53, and Bacillus aerophilus. And for your kind informations, all the other samples, the scientists, they deposited to Imtech Chandigarh and NCCS Pune, but these samples are not available. Very recently, some scientists from abroad, he wrote one article and he asked these samples from our repository, but the samples are not available of the other samples. But our samples, it is available with us, and we have recently submitted to NCCS Pune and Imtech Chandigarh also. So that's why this sample is very interesting. And we also went for the comparative genomics of this sample Bacillus altitudinus SORV11, and they showed the core genome and the pangenome also, and they showed the similarity and their dissimilar portions also. So like this way, to some extent, SORV is different with other. So these are the unique genes we found in our samples, and it is interesting for UV resistant and as well as exopolysaccharide production also. So then we went with the Bacillus altitudinus, the reference sample, and with our samples with the MOB alignment. So it is the multiple alignment of the conserved genomic sequences between these two samples. Here also we focused on the similar regions and the dissimilar regions, and very interestingly, although both are almost same, but their genomic orientation or arrangements is different. So this is prophage region. We also studied the prophage region of our samples with in comparison with the other samples, and the prophage genome similarity was similar with the Brevibacillus phage GMR1. And this is the in detail study of the prophage arrangements of the sample. And this is the class stage plot of our samples, and it shows different bins and the locations of each and every genes. So these are the comparison with different strain of Bacillus itself, and they are different genes. They are responsible and they are present in the genome. So UV repair mechanism, we studied spatially the different mechanism system. So we found DNA repair protein RAD that are present in all the strains, and DNA repair protein RADC, it is also present. So like this way, some protein they are common and they are present in all the samples. But some interesting DNA repair protein like RECT, this is only present in some samples, but not in all the strains of Bacillus. So this is the major finding of Bacillus altitudinus. We found the genome size we found more or less with the other samples is quite similar. GC content is almost 41%, and SORB showed 100% similarity with the type strain 41KF2B. Although their genomic rearrangements are different, we also calculated ANI of the sample, and a total of 3867 coding genes are annotated, among which 2 are rRNAs and 22 tRNAs are there, and 102 genes belong to DNA metabolism, out of which 55 are associated with DNA repair. So here is the interesting things, and we have planned to study in details with all those genes. Maybe that's why this sample is different, and their habitat also different with the others. We also studied the core genome and the pangenome of these Bacillus species, and more important finding is that the absence of recombinational DNA repair protein RECT was absent in both extremophilic strain SORB11 and 41KF2B. So this is one story. And other story is related with a sample that is Bacillus anthracis. So in the beginning, when we started the work, we isolated this sample and we went for 16S rRNA gene sequencing, and we found this sample is matched with Bacillus anthracis. As this is Bacillus anthracis, and we know from the literature, causal organism of anthrax is Bacillus anthracis. So in our university or in our laboratory, we don't have the facility to handle all these kind of samples. So we are worried. We are supposed to stop the research in the beginning. But I thought I should go for the testing of the sample, whether they are pathogenic or non-pathogenic. So I tried with the RAD model with our colleagues at our university in the geology department, but we cannot get success. The result was not promising, and they took long time. So meantime, we thought, why not to go for the whole genome sequencing, and we may analyze the data whether this is pathogenic or non-pathogenic. So that's why we went for the whole genome sequencing. And in this slide, you can see in North America, Bacillus anthracis, they reported 103, and from Canada it is 1. And from Asia, China reported 6, South Korea reported 2, Japan reported 1, and Israel reported 1. And from India, we reported only 1, and this is the study. And South America, Brazil, they reported 2, Argentina, they reported 13, and you can see the Europe, different country, they reported different numbers. But interestingly, all the Bacillus anthracis, they are pathogenic. Whatever the report are there from North America, South America, Europe, or Asia, all they are pathogenic. They cause anthrax. But interestingly, we got this sample Bacillus anthracis, but it is non-pathogenic. So it is very important. This is non-pathogenic. We tested first through the help of bioinformatics. That's why my title is that the bioinformatics can also tell you the lifestyle of the bacteria or the microbes. So here, with the help of Dr. Rajiv Bharsne from ICRISAT, we went for the whole genome sequence and we did the whole genome sequence analysis. So this is approx 5.1 MB in size, and GC content is 35%. Total gene is 5474. And these are the general informations of the whole genome of Bacillus anthracis. So when we went for the different analysis of this Bacillus, we check the different regions or different plasmids that are responsible for the pathogenicity. But interestingly, it was absent for our cases. So we named this bacteria as PFAB2. It is from Panifola. It's a hot spring water, and from that water we reported this bacteria. This is something different because this is not the pathogenic one. So they have different biochemical characters, responsible genes, and they have different other makeup. That's why they are not pathogenic. So these are the general informations about the Bacillus anthracis. So one genes that encoding for polygamma glutamic acid biosynthesis, that is responsible for the pathogenicity. But interestingly, this is absolutely absent in our strain. So that's why this is not pathogenic. Anyhow, further, I am not showing the detailed wet lab study with the help of Professor Bhatnagar from Delhi University, from JNU, right now he is Vice Chancellor of PHU. So we studied the wet lab in his laboratory. We had some collaborative work, and we found in wet lab also this is not pathogenic. So we analyzed each and every data of the whole genome, and we found some interesting genes. We found different regions. We went for the ANI heat map also with Bacillus anthracis, with related Bacillus anthracis, which cause the pathogenicity. And we found this is something interesting and quite different. So we studied the island view. In this island view, we can see the different virulence factor and their pathogenicity associated with the genome, and virulence genes is absent for this case. So we studied for pan-genomic analysis and the core genome analysis, and we also studied the FOD genes to see the different things. So our strains we compared with the highly pathogenic strain, that is Amaze ancestor. This was reported from South Korea, and here you can see the pattern or orientation. We have compared with A and B, that is A with our sample PFAB2 with the more pathogenic strain MS ancestor. So this MS is available at JNU. We did this work there, and we reported our sample as non-pathogenic one. So this is the detailed analysis and study different genes which are responsible for different pathway and EPS production also. So this is our major finding of the sample. The worldwide near clonal spread of the organism suggests that expression of the toxin and capsule genes confers an advantage of Bacillus anthracis. Thus, complete absence of capsule biosynthesizing gene in PFAB2 can be a probable explanation of the spread of non-pathogenic variant of Bacillus anthracis from extreme environment to natural other surroundings. So Professor Bhatnagar told us during our study that it is the common phenomena for some bacteria. The non-pathogenic may be converted to pathogenic, or some pathogenic may be converted to the non-pathogenic to pathogenic, and in nature, if it happens, it may take long time, say more than 20 years. But for our cases, we got this thing as a non-pathogenic. So we are planning to study more in the wet lab for the conversion of this kind of Bacillus anthracis. And other important characteristic factor for virulence in pathogenic Bacillus anthracis is the presence of poly-gamma D-glutamic acid capsule. This capsule is important for its survival in host, thereby evading the host immune system. By contrast, genes encoding for poly-gamma glutamic acid biosynthesis are completely absent in PFAB2 strain. So this is the two stories we tried to solve with the help of bioinformatics, and for this work we get financial help from Ministry of Art Science, NCOR Goa, and earlier I used to work with BIT Mesra Ranchi. Now I am at Badwan University, and Dr. Rajiv Bhatsne from ICRISAT, he helped us. We also get help from Navi, Dr. J.K. Roy, and Imtech Chandigarh. In collaboration with Chandigarh, we did this work. So this is our group. Aparna Banerjee, right now she is in Chile. She worked with Bacillus anthracis, and Urmi, she worked with Bacillus altitudinus, And other scholars, they are working with different microbes. Mostly they are extremophiles from volcanoes, from different industry, and we are working on antibiotic also. So this is the things. So thank you all for your kind patience and listening to me.
Thank you, Rajiv sir. Hello.
Yeah.
Thank you.
Any question from
Can I ask you one
attendees?
question?
Yes, yes.
Vijay, you want to go ahead?
No, no. Ask question.
Okay, all right, fine, thank you. Okay, Dr. Rajiv, I have one question. Very interesting, these bacteria have these things. I'm just wondering, I don't know, I know there is some research going on this tardigrades. You might have heard tardigrades.
Yeah.
So I'm wondering because, while I was coming to know about it, that is also resistant to UV and it survived, so many eras during evolution, it did not get extinct easily and I think it survives UV. I was just wondering if you are aware of anybody is trying to look at the bacteria that could be inhabiting in that one, and is there any so similar kind of genes found there also which gives UV resistance to that tardigrade, which
Yeah.
not many organisms, which is definitely not a microbe, which is a very tiny though, but I'm wondering if you had any idea about that by any chance.
Actually, we compared
Hmm.
with other sample, that is Bacillus endurance. This was the first reported bacteria which can resist UV, highly UV resistant. So we are comparing with only the bacterial system and not with the other one.
Okay. Okay.
So
Thank you.
things is going on. We are
Yeah.
working on protein expression level also.
Yeah, but yeah, there is not a lot of work on tardigrade, but I think I don't think sequences have been available yet. But I was just wondering if there's people are trying to isolate at least bacteria out of it. Perhaps
Yeah.
not yet, I think. But yeah,
No.
thank you for that answer.
Yeah. Yeah.
Hello. Any other question? So thank you. Our next speaker is Dr. Harpreet Singh. Dr. Singh is currently working as assistant professor of bioinformatics at Hansraj Mahila Vidyalaya, Jalandhar. He has more than 10 published articles in national and international reputed journals. He has more than 10 years of teaching and research experience in the field of bioinformatics. He is also an active core member of Bioclues organization and currently serving as a finance secretary. He has also recently been nominated as an executive member for AP Bionet. So today's topic of his talk is deciphering group V1 serpins from uh, sequence variation structure and evolutionary perspective. So Hafiz sir, please.
Respected yes, most respected Dr. Professor P.K. Gupta ji, Dr. Vijay Kumar Gehlot, all the members of the organizing committee, especially Dvendra Kumar Biswal, Pras, Subuntu Vure, and the president of Bioclues, Professor Narhari Shastri, and the former presidents and most respected Dr. Jayaraman Mulati. A very good afternoon to all of you, all the dignitaries, as well as all the participants. It's a great privilege for me to be a part of this third INVIX conference as a speaker. I have been associated with INVIX as a core committee member, as a program chairs, but this is a new experience for me, just to share my experience, my research experience uh, with the diversity of audience here attending this prestigious conference organized by Bioclues organization. Being a part of Bioclues core committee, I'm really honored to be a part of this conference. I will be sharing my views on the topic, the diversity of plant cyclophilins and their status in the wheat genome. and it's very relevant to the session on plant genomics, advances in plant genomics. So just to start with the cyclophanes, why our group is working on cyclophane, what fascinates us to work on cyclophanes, that has been summarized by us in our recent research article, which published in the end of September 2020, just a couple of months ago, in Frontiers of Plant Science, and with the title Plant Cyclophane, the Multifaceted Proteins with Versatile Roles. We have tried our best to compile the information about cyclophanes, particularly related to the plant cyclophanes in this manuscript, in this review article. Okay, so before going further, I just want to summarize what are the cyclophanes and why we are interested in studying cyclophanes in plants. And all of us aware that amino acids are the basic building block of proteins, and individual amino acids, they are linked together by peptide bonds. And most of the peptide bonds, they uh, prefer to have the trans conformation. But problem arises whenever there is a proline reduce in the amino acid chain, in the peptide chain, because the proline tends to attain cis conformation as compared to trans conformation. However, the trans conformation is important to be the proper for the proper folding of the protein, for the proper functioning of the protein. Therefore, it is very important to somehow convert this cis conformation to the trans conformation. Many of the proteins randomly adopt the trans conformation in the peptide bond preceded by the proline reduce. However, a majority of the the proteins, they require the intervention of a special type of enzymes, which are called the peptidyl prolyl cis trans isomerases, which we call as the PPIases. PPIases has been uh, categorized into various classes like cyclophanes, FKBPs, FKBCs, parvulanes, PP2A activators. Cyclophanes and FKBPs are the two major classes of PPICs, and they also are clubbed into the category called immunophanes, because both of them, they bind to the immunosuppressive drug. Cyclophanes specifically bind to cyclosporin A, while uh, FKBPs, they bind to the FK506 or rapamycin drugs. FKBCs are unique because they possess the domains responsible for binding of cyclosporin A as well as the FK506 drugs. So they have the dual function of having the properties of FKBPs as well as cyclophanes. Other two type of PPACs are less common, but they have been playing an important role in various aspects of plant cell and function. This is an overview of cyclophanes, which have been, you know, have been demonstrated, which have been identified in various organisms, ranging from E. coli to plants. And I think it's the second table is more elaborate, which gives you the overview of cyclophanes, genes, and proteins, and their important properties across the genome in bacterium, fungi, protozoa, animal, and plants. If you see in this diagram, although the cyclophanes have been present in diversity of organisms, almost all organisms they possess cyclophanes, the plant genome has been shown a great expansion in the cyclophilin gene family. And this has been attributed particularly to the recent genome sequencing experiment, which has been unraveled various plant genomes, including wheat, rice, cotton, brassica, and so on. There is a large diversity of cyclophanes, in particularly when we talk to plants. In plants, the lowest number of cyclophanes are present, genes are present in Oryza sativa, while 29, while the largest number of genes they are present in Brassica napus 94. The size of the amino acid is also a variable. If you go for the molecular weight and size, the lowest cyclophenol, the lowest molecular weight is 5.7, while in Brassica napus, while the highest one has been demonstrated, have been reported in wheat with the weight of around 385 kilodaltons. The PI range also varies a lot, varies from 4.4 to around 12.60 across various plant species. Cyclophyllins, they are characterized by a presence of a particular domain, which is called the cyclophylline-like domain. We will discuss the structural aspects of this domain later on. But this domain is mainly responsible for the PPI's activity exhibited by various cyclophylins. Apart from this cyclophylins, there are cyclophylline-like domain. There are many of the cyclophylins which possess additional domains, and that's why they are called the multi-domain cyclophylins. So these two tables give the contrasting view of the single domain as well as the multi-domain cyclophylins which are present in various organisms. In addition to the diversity of the domains, we also found that it has also been very well established that the cyclophylins, they are localized to almost all the organelles, all the cellular compartments, whether chloroplast, cytoplasm, mitochondria, endoplasmic reticulum, nucleus, and so on. They have been present everywhere. That's why they are ubiquitous in nature. They are ubiquitous in terms of the diversity of the organisms that possess them, and they are also ubiquitous in terms of the type of cellular compartments they reside in. And this is a particular example of the multi-domain cyclophylins, which we have shown a comparative analysis of cyclophylin homologs in Arabidopsis, humans, and yeast. Uh, the green shaded boxes, they represent the cyclophylline-like domain, while the other colors, they represent various different type of additional domains which are present in cyclophylins, including TPR, RRM, WD40, U-box, and so on. These domains have been very important in imparting additional functions to the plant cyclophylins that can be attributed to the plant pathogen interactions, seed development, regulation of embryogenesis, plant defense, oxygen signaling, antifungal activity, and so on. I have listed some of the important roles in which cyclophylins have been involved. For example, growth and development in plant, protein folding, hormone signaling, pathogen response, regulation of transcription, and stress adaptation. These are the few of the major roles which have listed here. I particularly want to focus one important role which we have again demonstrated graphically in the paper I mentioned we published in the Frontiers in Plant Science. And this the diagram shows how cyclophylins are important in regulating the auxin response gene, and they try to control how different types of cellular mechanisms they are controlled by the level of oxygen. This figure had two compartments. The upper compartment shows the oxygen absence of oxygen, and the lower compartment shows the presence of oxygen. And the important one is the key player in all these mechanism is the OXIAA set of proteins. The OXIAA set of proteins have a characteristic motif called the background motif, which has the presence of the important key residues like GP, GWPP, V. We will show how these residues are important because the proline is present here, and that's why the cyclophylins are important in controlling the expression of the genes. This OXIA in alone or in combination of the another partner TPIL, they bind to oxygen response factors in the absence of auxins, and they actually stop the transcription of the upstream genes regulated by the ARFs. In the presence of oxygen, what happens in that there is a protein complex this auxin binds to one of the key member of the protein complex called TIR1, and this TIR1A combination, then it binds to this OXIAA at the diagonal motif. But this binding is very peculiar in nature. Peculiar in nature means that OXIA binds only to this, diagonal, the TR and IA complex when this diagonal motif is having the peptide bond, a particular peptide bond between P and B in trans conformation. Normally it is in cis conformation, but it must be in the trans conformation. This cis to trans isomerization is very important, and that cis to trans isomerization is controlled by a cyclophin called OSIP2, which have been identified in Rhiza sativa. Once this binding, is there due to this cis to trans isomerization, what happens is that this OXIAA group of protein, they are ubiquitinated and they are degraded by 2S proteasome machinery, and that led to the expression of the auxin responsive gene, including the OXIA. So this is a feedback mechanism actually. So this is how the cis to trans isomerization is important, and how cyclophilins are playing important role. This is a typical example showing the importance of cyclophilins. Having said, given you some idea about the importance of cyclophilins, their ubiquitous nature, the diversity of roles they are involved in, now let us come to the second part of this talk, and that's how I titled it at the motivation part two, because we published our work on genome-wide analysis of cyclophilin gene family in wheat, and we also published how there are some of the cyclophilins, they act as heat stress. They act in response to the heat stress in plants, and that was possible only because of the publication of a very complex wheat genome in data 2018. And when we explored the wheat genome for different type of cyclophilins, we found that the wheat cyclophilins in wheat, there are around 83 different cyclophilin genes that encode for 85 different proteins. Actually, 89 proteins were there, but there are 4 of them were the identical copies we found. Interestingly, in all the 21 wheat chromosomes belonging to the 3 different wheat genomes, cyclophilins have been distributed in all the genomes. With the maximum number of cyclophilins, they were concentrated on the 7 chromosome number 7 in all the 3 genomes, 7A, B, D, and the minimum number of cyclophilins were found in the chromosome number 1A, 1B, and 1D. We also find many of the cyclophilins which were duplicated in nature. The different color combination, they show the homologs, cyclophilin homologs, because wheat is a combination of 3 different genomes, and due to the presence of 3 different genomes, most of the genes are present at triplicate. That was what we expected in our study, but we also found some surprises. For example, many of the genes, they did not show this triplet trend, but they were either lacking some of the partners, or they were having some additional partner due to some duplication events. And the smallest cyclophilin in terms of amino acid range from 160 amino acid, while the largest one having 823 amino acids. So the trend of the variation in pi values we observed in all other cyclophilins, that was evident also in wheat cyclophilins. That additional copies of cyclophilins, in addition to the triplets, they have been attributed to the many duplication events which we showed in form of a Sitko's plot in terms of the segmental duplication, the tandem duplications, and ectopic duplications. We also try to analyze how the gene structure of the cyclophilin varies, and we very interestingly we found that there have been a lot of variation in the number of introns in the coding regions as well as in the UTR region. Just as per the table shown here, there were 13 genes which were devoid of introns. They were not having an intron. Interesting, in these 13 genes, there were many of the genes which are duplicated genes which were not having the intron. They were devoid of introns. Eight, eight of the genes were having single introns, while three of the genes were having the maximum number of introns, that is 13, uh, succeeded by six genes which are having 12 introns. Although most of the duplicated introns were devoid of duplicated genes were devoid of introns, but there were some of the genes which are having additional, which are having intron instead of their triplet homologs, and there were also some of the genes which the triplets were having introns, but their duplicated value was devoid of. So that type of variation we observed in terms of the gene structure. We also found a lot of variation in the size of the intron. The smallest intron uh, was uh, having 73 uh, having 73 base pair, while the largest intron was having around 28.6 kilo kbs in size. In addition to the introns in the coding region, we also found that there were uh, there was the evidence of having introns in the 5' and 3' UTR regions in different wheat genes. As per the subcellular localization, most of the cyclophilins were distributed in cytoplasm and chloroplast, followed by nucleus and the mitochondria, as well as some other organelles. Means almost all the organelles, plant organelles, they showed the presence of different type of cyclophilins, different uh, type of cyclophilins, and different number of cyclophilins in them. As we just mentioned in the previous overview of the cyclophilins in various organisms, there many of the cyclophilins, they possess additional domain in addition to the cyclophilin-like domain. In wheat, out of the 85 different cyclophilins, 58 cyclophilins were single domain. They were only consisting of the CLDs. However, there were many cyclophilins. They were having some additional domains, including the classical U-box domain, RRM domain, WD40, TPR. We also found that there are two unique cyclophilins in weight, which were having the F-box domain and PAN domain. These domain has not been earlier detected in any of the cyclophilin gene family studied across different genera. These domains have been able to give a different attitude, different functions to these cyclophilins, including involvement in the protein-protein interaction, protein folding, splicing, signal transduction, and so on. We also try to analyze how they are related phylogenetically. Different cyclophilins are related phylogenetically, and what we found that thus in the phylogenetic tree, a trend was shown that cyclophilins, they have been clustered together according to their localization signals as well as according to the domain organizations. Means the cyclophilins which are localized to a particular organelle, for example, the red color nuclei, they are clustered together, and also they were clustered together. Additional clustering was done based on whether they are single domain cyclophilins or they are the multi-domain cyclophilins. The cyclophilin domain structurally is actually a very unique domain consisting of 8 beta parallel sheets. They have been intervened by the two important helices, alpha helix 1 and alpha helix 2, and they form a hydrophobic surface on which the direction of cis to trans isomerization take place. All the secondary structural elements, they were found to be conserved as depicted by this plot. All the secondary structure elements, they were shown to be conserved among most of the cyclophilins. We also noticed some of the variations, some of the deletions, insertions in some of the important parts, but most of these variations were limited to uh, the non the core structural part of the cyclophilins. It, in addition to the involvement of cyclohexylins in different cellular functions, we also found that many of the cyclohexylins, they respond to the redox potential, they respond to the oxidation reduction mechanisms in plants. And there are two type of behavior which have been shown in terms of the redox regulation. The one is the direct mechanism in which what people have reported that two important cyclophilin cysteine residues, 16122 and 16126, which are very, which are the part of the core, the active site of the cyclophilins. They, when they form the disulfide bond on reduction, they actually, led to the closure of the active site very quickly. They led to the closure of active site, and the PPI's activity is lost, uh, just in a fraction of seconds. In addition to this mechanism, some of the cyclophilins has another redox mechanism in which the cis40 and 6168, they, when they make hydrogen, they make a disulfide bond between them on particular conditions, they do not immediately lead to the closure of active site, but they led to the closure of active site through a indirect allosteric mechanism, through a process called conversion loop mechanism. What we found that there is a third category that have been published in our one of our previous publications, that TSIP A1, that is very unique and that possess both the mechanism, that possess direct mechanism, that possess the indirect mechanisms also. That in that sense, TSIP was unique. So this table also demonstrate the status of different cyclocysteine residues in different cyclophilins. Like the number of cysteines range from 0 to 7 in different cyclophilins, and it indicate that there may be many other cyclophilins which which are controlled by different redox mechanisms in different ways. So these are the figures which show the distribution of different mechanisms. Like the left one shows, which have been found in TSIP 17.4, the presence of the direct the residues involved in direct mechanism like cysteine 122 and 126. The right one shows the presence of allosteric mechanism involving C40 and C168. And in the next slide, there have been TSA 21.1 and 23.2, which are showing both the presence of direct as well as the indirect controlling system residues. Apart from various cellular functions, we were also interested in finding, are there some of the cyclophilins which take part in different stress response in wheat. So we try to try to generate a heat map of cyclophilins from the gene expression data, whether to know whether there are some of cyclophilins which are involved in heat stress. And what we found that there are some cyclophilins which have been shown a great contribution imparting resistance or tolerance to heat stress to different plant species, and particularly in wheat. We also did some experimental analysis. We showed experimentally that there are the cyclophilins which are involved in heat stress response. But for the shortage of time, I haven't put that slides in this particular presentation. Just to conclude, we can say that in wheat, there are 83 different wheat cyclophilins which encode for 85 different proteins, ranging from 70 to 92 kilodalton in their size. They are distributed all the 21 chromosomes, in addition to the three homologs, typical the triplet variation trend. We also found that there are some of the cyclophilins which the homologs which are having additional copies, and there are some of the homologs which are devoid of the triplet trend. Cyclophilins are single domain as well multi-domain, and we also found that we found some novel cyclophilin domains like F-box and PAN, which has not been reported in any other cyclophilin gene family. CLD is very conserved as a typical feature of the cyclophilins, and cyclophilins have been localized to various organelles. We also did the protein-protein interaction network studies, and also try to understand, are there some specific cis-regulatory elements in the upstream region of different cyclophilin transcribing genes? And we found that we found many signals which suggest that cyclophilins interact with a variety of proteins controlling different type of cellular functions, and also there are some specific cis-regulatory elements which make them specific for the expression during different type of cell response like hormonal, plant pathogen interaction, and other such abiotic and biotic stress responses. Having this analysis of this data, we found many candidates which are very interesting in nature, like many of the multi-domain cyclophilins, many of the cyclophilins possessing variation in the cysteine residues, which we are exploring at experimental level to find how they behave in different experimental conditions, how their structures look like, and we are also in a position right now to purify and crystallize some of the multi-domain cyclophilins so that we can tell to the audience how multi-domain cyclophilins behave in wheat, and that's how in other plant species. So this is what we are planning to do in future. I have summarized some of the future projections, but I will not uh, I will curtail my discussion just to save time. Yeah. These are some of the publications which we have generated from our different studies in cyclophilins, and this entire work has been a team effort under the leadership of Professor Prabjit Singh, Professor, Department of Biotechnology, Guru Nanak Dev University, Amritsar, and my teammates Supreet, Kiran, Mangal, Gundeep, Tanima, Harsimran Kaur for their help, for their contribution in different aspects of the studies on cyclophilins, and many of the different things we are exploring at lab. These people are playing important role in the experimental generating experimental data and doing experiments, different type of experiments. This entire studies have been funded by different funding agencies, including DST, International Wheat Genome Sequencing Consortium, Department of Biotechnology for doing the experimental part. So for these reasons, I am thankful to all the contributors, all the different stakeholders, and I also thankful to the Bioclues organization for giving me this opportunity, and not only this opportunity, was also helping in planning different studies related to the cyclophilins also. And at the end, I'm thankful to my college, Hansraj Mahila Mahavidyalaya, and the DAV College Managing Committee for providing us opportunities to work in such organizations like Bioclues, and also to work with other collaborators to do some to get some insight into the world of plants. And I'm thankful at the last to the organizers, all the organizers of this great conference in BICS 2020. And with these words, I will stop my presentation, and I will be really happy to have any queries, have any questions, as well as also having some suggestions, because the chairperson of this particular theme, they are themselves the stalwarts of plant biotechnology, plant breeding. So I will be happy to get some suggestions from them also. Thank you very much.
Hello. Thank you, Arpit. Any questions from any attendee? Hello.
Good afternoon to all.
Yes, Harpreet. Can you hear me?
Harpreet, your microphone is muted. Harpreet, your microphone is muted. Yeah.
Muted it now.
Okay, okay. Now I think my microphone is unmuted. Yeah.
Can you hear me, Harpreet?
Good evening, sir. Yes, good afternoon, sir. Sat Sri Akal.
I want to know whether cyclophyllin can be used for developing heat tolerant heat phenotype.
Yes.
Harpreet, did you get the question? Can you hear me?
Yeah, Harpreet, sorry to interrupt you. Sorry to interrupt you, sir. Harpreet, we cannot hear you, please. It's not audible from your side.
Muted my microphone.
Are you able to hear me now?
Yes, yes, the voice is low. Vijay, would you please look into this? Yeah, thank you. Yeah.
Can you hear me?
Hello. Is my voice clear now?
Yeah. Now it's clear.
Yes.
What about
Yeah.
mine?
Now you're clear, sir. I don't know. There was some disturbance in between. Now everything is clear. So please ask your question. Yeah.
I want to know whether cyclophyllin can be used for developing heat tolerant phenotype of wheat.
Sir, actually, that's what we are trying to ask some questions. We have identified some of the candidates, and in the first stage, and now we are moving in the second stage where we are trying to clone them, and we are trying to see what type of and what level of heat tolerance they show. In the initial RT-PCR analysis, they have shown some evidence, but we want to clone and we want to do some detailed experiments. Yeah.
Vijay, I think we are already late. Therefore, we can proceed to the next speaker. Oral presentation.
Uh, uh, Hello.
Hello. Yeah.
Thank you, Arpit, for excellent presentation. So no more question is there. So we move to oral presentations, and we have in this session we have 4 oral talks. First one by Anil
Okay.
Kumar. So Anil, please.
Good afternoon to all.
Thank you very much. Thank you.
Working as assistant professor in the Department of Biotechnology, Vignan Foundation for Science, Technology and Research, Deemutubi University, Andhra Pradesh. At the outset, I would like to thank the organizer for giving me this opportunity, especially Dr. Prashant and Dr. Subnakar.
But keep to time. We are already late. So 8 minutes.
Genome-wide identification and characteristic profiling of small heat shock proteins, gene family under various diverse abiotic stress conditions in sorghum bicolor. As we all know that plants lack locomotion, so they need to be stay in that particular place. Due to their sedentary nature, they always face harsh environment conditions and they need to live in the same environment. Of the various abiotic stresses, heat is one of the most important abiotic stress, which results nearly 70% yield loss. In order to cope up all these stresses, plants have developed different mechanisms so that they can tolerate the different abiotic stresses, particularly heat stress. And in the today's topic, I told you I'll talk about heat shock proteins. First of all, what are heat shock proteins? Small heat shock proteins are the proteins which are referred to the small in size, from 12 to 42 kD. Whereas heat shock proteins are those proteins that are expressed under heat shock conditions. And some of the small heat shock proteins are also reported to form HSP60 and HSP70, which means that these are the bigger heat shock protein families. And in the present study, we had mined all the small heat shock proteins that are there in sorghum bicolor. As we all know, sorghum is a drought resistant crop, so we have chosen that crop, and we have mined all the genes that are responsible for small heat shock proteins. Yeah, straight away coming to the results, we had mined almost 47 genes from sorghum, and this particular slide represents the chromosomal localization of various heat shock proteins. And in this, some are represented in similar color, okay, color wise, they do come under segmental duplications, and this particular lines are the barred lines, they represent the regional duplications. So the location of SPHS has been found on all the chromosomes. And in this slide, it explains about the gene characterization, wherein you can see the presence of CDS portions followed by upstream and downstream, wherein we can see the exons are around 1 to 5 in all the 1 to 47 genes, and this is about the conserve motifs, wherein we had identified the signature sequence in all the respective genes. And they are the similar signature sequences, they are represented in similar color like red, violet, blue, all these things. So from this, it is clearly evident that they do contain the domain, heat shock domain. Because of them, they are grouped under this family. And this is the phylogenetic analysis of 47 SBHSP genes, wherein they are falling into different clades, okay, and whether they are small heat shock or not, we had identified based on the presence of their size, and all of them are in the respective size only, 12 to 40 kD only. And in this, we have done the phylogenetic analysis in comparison with Oryza sativa, Arabidopsis, and sorghum bicolor, wherein we identified they are pulling based on their structure, function, and localization. And this slide represents the digital expression of all the heat shock proteins. We are done with the help of gene investigator, wherein we identified some genes are highly expressed and some genes are very and low level expression has been reported in some of the genes. And similarly, this is the tissue specific expression level, and this is the developmental level. Later on, in order to analyze the genes that are highly expressed in digital expression, we had chosen those genes like 2, 7, 9, 15, 20 up to 47. We had chosen the genes which are highly expressing in digital, and we worked out in the in vitro condition with the help of qRT-PCR, that is nothing but real-time PCR, wherein we identified some genes like HSP2 is highly expressed in almost all the conditions like broad root, salt root, salt stem, salt leaf, heat root, heat stem, heat leaf, cold root, cold stem, and cold leaf. Whereas some other genes like SBHS7, they are least expressed. So based on their transcript expression level, we had drawn this heat map wherein some genes are highly expressed and some are, some expression is very low. And this is the first part A picture represents the native expression of these genes in root, stem, and leaf. And below the diagram represents under various abiotic stress conditions like drought, salt, heat, and cold in different tissues like root, stem, and leaf. And the entire work has been published in International Journal of Biological Macromolecules, and this was published in the year 2020 in the January edition. And Mr. Nagaraju, he is the first author of this paper. Coming to conclusions, these conclusions, based on the present work, we have derived the following conclusions. We have reported 47 SBHSP20 genes, and we found very interesting thing of 47, 17 are localized to chloroplast. Based on their expression analysis, based on their digital expression followed by the real-time expression, we found that these genes are not only expressed in particular to heat, but they're also expressed to salt, drought, and cold tolerance also. We can use these genes, I mean the genes that are highly expressed, for developing new transgenics for different abiotic stresses. Coming to acknowledgements, I would like to thank Dr. M. Nagaraju, who is the first author of this paper, and Dr. Palakulan Sudhakar Reddy, who has helped in doing real-time PCR, Dr. Anish Kumar, helping in digital expression, and Professor P.B. Kavkishore sir, who is my mentor and who is the corresponding author of this paper. Under his able guidance, we had finished this work. And I would like to thank everyone. If any questions are there, please mail me. I can reach out at anilkumar.ou@gmail.com. Thank you. Thank you, one and all.
Thank you, Anil. Anil, are you there? If Anil is not there, so we can move to next oral talk by Pradeep Phule. Pradeep?
Yeah, so Pradeep is facing internet issue, but I have his video with me. I can play with my computer, Abhishek.
Okay, okay.
He will be available for answering the questions.
Okay,
so
please.
let me first play the video. Okay.
Yeah, if it is okay, I'm airing it from here, Abhishek.
Yeah,
Is it fine?
it's okay. Yeah, you can do it. Good. Thank you.
Yeah. Sorry, 1 minute. Yeah, I'm lost. Yeah, I'm sorry, 1 minute. Really sorry, 1 minute.
Primer, my topic is towards the discovery of secondary metabolite gene cluster in novel marine fungi, Pygeron species PP1-1. I started my PhD work by collecting various marine samples from the Baltic Sea. Fungi were grown into the lab, and those fungi which were growing into the salt-containing media were considered as a marine fungi. Using the ITS primer, I amplified using the PCR technique and sequenced using the Sanger sequencing. And using blast analysis, we identified closely related species to it. As you can see, the circular pattern growing fungi, this fungi was selected for the Illumina sequencing. This assembly was done by the CLC Bio and AB software, and these two assembly were merged together into the 42.46 MB of genome. It has a very good N50 value, and it was assembled into the 411 scaffolds. Using Augustus, 13,162 protein coding genes were found. Using maximum likelihood tree of 36 Fusarium genus, which were collected from the NCBI, the phylogenetic tree was drawn using the RAXML VTR gamma model. It was found that it is closely related to the Fusarium solani and Fusarium tristinctum, which were soil-originated fungi. Using repeat masker, genome of Fusarium PP1-1 consists of the 1.02% of the repetitive region, in which 0.14% were LTR elements gypsy, and TC1 POGO elements were 0.18%. I designed a pipeline program using Signal P, transmembrane Markov model, Target P, and Volt P sort softwares. And I found 1,109 proteins were predicted as secreted proteins, which were 8.38%. As compared to the other fusarium species, this number of secreted proteins was quite less. In gene annotation and pathway mapping, I found in KEGG pathway 360 sequences encoding 139 enzymes which were involved into the biosynthesis of antibiotic pathway. In InterpoScan analysis, most of the sequences were major facilitator superfamily, short chain dehydrogenase, and cytochrome P450. Using Augustus, I did a secondary metabolite gene prediction. I found 60 gene clusters with 1,442 genes. That is a 10.9% of total gene content were involved into the secondary metabolic gene cluster production, in which out of this 60, 28 gene cluster has shown greater than 30% similarity with other fusarium gene clusters. As you can see in the table, 18 gene clusters were NRPS, 15 were T1PKs, and 9 terpenes. Total were 16. Here in the unique number is shown into the bracket. These genes were only present into this fungi. Here in the heat map, you can see the white color represents unique to the fungi. That is, it is absent into the other fungi. Here for comparison, 4 representative of the fusarium were taken. Fusarium fusiculae, Fusarium graminum, Fusarium obsisporum, and Fusarium verticillate. Yellow color represent the intensity of the gene. That is identical, how much identical these genes are. The more the yellow color is, more is the identical, and the lesser the yellow color is, the less identical. In the left hand side, you can see the type of the secondary metabolite and whether it is present into this fungi or not. We did its transcriptome analysis into the 4 different conditions. For this, we did co-culture experiments, and we found that using the Bacillus subtilis and metabacterial sample, it is growing very faster. So in this study, metabacterial condition is a growth of the fungi from the same place we collected the bacterial sample. So we named it as a metabacterial sample. So we mainly consider 4 conditions. Using these 4 conditions, we sequenced its RNA, and using RNA-seq, we did its transcriptome analysis. In transcriptome analysis, this is the Venn diagram of all differentiated regulated transcripts, p-value 0.05 and log fold change greater than 2. As you can see, salt versus no salt condition, salt versus Bacillus subtilis condition, salt versus metabacterial condition, and Bacillus versus metabacterial conditions. So you can also see the down regulation and up regulation. Here you can see 458 genes were only specifically expressed into the salt versus no salt conditions. 471 were salt versus Bacillus subtilis conditions, while 168 were Bacillus into the metabacterial conditions were expressed. Here you can see the salt versus metabact and salt versus Bacillus conditions has the most of the common down-regulating genes. Similarly, they have the most of the common up-regulated genes. We also drawn a heat map of expression of core secondary metabolite transcripts in different conditions. Here you can see the cluster 1 is specifically expressed into the Bacillus and metabacterial condition. That is, these genes, this suggests that these genes are specifically expressed into the co-culture conditions. That is, cluster 1 is expressed into the only co-culture condition, whereas cluster 3 is specifically expressed into the salt condition, and cluster 2 has without salt condition. We also check the expression of cytochrome P450 and transporter encoding transporter encoding transcript. These are important for secondary metabolic production. So as you can see, the cluster 5 is specifically expressed into the no salt condition, cluster 3 in salt condition, and the cluster 1 in metabacterial condition. Similarly, you can see the transporter encoding transcripts. The cluster 5 is specifically expressed into the no salt condition. Cluster 4 is specifically expressed into the co-culture conditions, whereas cluster 1 is expressed into the salt condition. After that, we also sequenced the biodata from the same fungi. Using Kanu, I got 176 contigs. And using Hulk, I did error correction, and LRSCAP, I did its assembly into 114 scaffolds. And then these two assemblies were merged into the using quick merge. I got 54 scaffolds. Basically, I had the data of the Illumina assembly, that is the Physarum genome version 1, which were assembled into the 411 scaffold. These were together combined into the LRSCAP. And finally, I got 54 scaffolds, which is quite close assembly. At the end, in summary, different fungal isolates from marine origin were isolated. Genome of the Physarum species pp1-1 was sequenced and annotated. We did its growth test, and we found that in salt condition, it's showing higher growth. We did its co-culture under co-culture condition also. It has shown good growth. In anti-smash result, it has shown presence of higher number of NRPS, PKS, and terpenes. And in transcriptome analysis, Physarum species pp1-1 in salt versus no salt condition and culture condition shows significant expression changes. Finally, I would like to thank Professor Dr. Frank Kimken, who is a supervisor working in Christian Albert University, Kiel, Germany. Dr. Abhishek Kumar, who is currently working in Institute of Bioinformatics, and lab member Hannah Smith and Dr. Christina Colette-Lice. Thank you.
Thank you, Pradeep. Pradeep, are you there?
My topic is towards the discovery of secondary metabolite in novel marine fungi, species pp1-1.
Hello.
I started my PhD by collecting various marine samples from the Baltic.
No, any questions from anybody?
Good afternoon to all.
Hello. Hello. Any question? If no question, we move to next oral talk by Omkar Kulkarni.
Myself, Omkar. Here, I am going to present our work regarding genome sequencing of halophyte Salicornia brachyata. So it all started with one of the major problems in agriculture, that is soil salinity. Salinity stress affected over 6% of the area, and area is increasing day by day. This problem affects productivity, microbial community, and in turn the agriculture economy. In such case, of salt-tolerant plants can be beneficial. Such salt-tolerant plants are called as halophytes. Halophytes can complete their life cycle in high saline conditions. Therefore, it is important to study the evolution and integral salt arrest mechanism. In spite of having significant qualities, genome information for halophytes is as of now not accessible. Talking about halophytes, Salicornia brachyata is an extreme halophyte belonging to the family Amaranthaceae. This plant can accumulate salt as high as 40% of its dry weight. It can be cultivated for extraction of edible oils and eaten as salad greens. Therefore, we decided to use Salicornia as a genomic research to study salt-tolerant mechanism. We estimated the genome size using flow cytometry, which is to be 480 to 500 MB. Now let's look at the data generated for the de novo genome assembly. We generated short read data using Illumina HiSeq platform and long read data using PyBioSQL platform. The data summary statistics is as below. Here we can see the coverage for genome assembly is considered to be good. Here we generated two types of assembly. First is long read assembly using only long read data. For this we used Kanu assembler. And the second assembly is the hybrid assembly for which we use short read data and long read data both. This assembly we generated using Masurka tool. Here for assembly assessment, we will be looking at three. First is N50 value. N50 value basically gives us idea about the contiguity of the assembly. More the N50 value means more contiguous is our assembly. Second parameter is the Busco completeness analysis. Here the Busco checks for the core genes in our assembly. So the Busco score more than 90% is considered to be good. And the third thing is mapping with short reads. The short reads from Illumina are remapped to our assembly to check how much data is covered in our assembly. So if we see here, our hybrid assembly from Masurka assembler has N50 of around 10 MB and number of contigs to be 469. And if we check for the long read assembly, the N50 value is of 20 MB and number of contigs are higher than the hybrid assembly, which are around 1300. For both the assemblies, Busco score and the mapping rate are above 90%. Then we merge these two types of assemblies, that is long read and hybrid assembly, to get a merged assembly. If we see the assembly assessment of this assembly, then we can see that now the N50 has increased to 28 MB and number of contigs are decreased to 409 and other parameters are similar. Now we used optical mapping data to super scaffold this contig level assembly. For this, we used BioNanoSolve suite. After optical mapping scaffolding, here we can see a drastic change in N50. It's gone up to 60 MB and the number of contigs are as less as 340. And also the Busco score is above 90%. Then we just polish the assembly using our highest quality short read data, and there are some slight changes in N50 and Busco score. To summarize all these things, as we go on refining our assembly, we got the N50 value to be increased and the number of contigs to be decreasing. So the Busco score gives us idea about the coding part of the genome, while assembling the repetitive part of the genome is the challenge for any genome assembly. So for that, we calculated LAI index for this purpose. LAI is linearly correlated with the content of intact LTRs identified in the genome. So for our assembly, we got LAI of about 14.29, which if we compare with the table above, which is of reference quality. So what does it mean? It means that intergenic and repetitive sequence space is also assembled well with reference quality. Then we move to repeat annotation. For this purpose, we use repeat masker tool. We got around 65% of the genome covered with the repeat sequences. Then we compared the repeat distribution with the closely related species. And here we can see that the distribution is also similar. Then we moved on to the gene prediction and identification. For this purpose, we generated isoseq data as an evidence for gene identification. From this isoseq data, we generated transcripts from isoseq3 pipeline and given it to the maker2 pipeline for gene identification. Further, we are planning to generate RNA-seq data, which will supplement to these annotations, and also we'll be planning a differential gene expression analysis of at different saline conditions. To summarize all these things, we generated high-quality de novo genome assembly of size 469 MB. The assembly is having high N50 value of 60 MB and Busco completeness score of 92%. As we discussed, LAI score of 14 means reference quality assembly. The genome contains around 65% of repetitive genome, and the gene identification analysis is still going on. Thank you all for your attention. This entire project is a huge collaboration between CCNG and CHM and CRI. I thank all for this. Thank you.
Thank you, Omkar.
Yeah, am I audible?
Yeah. Omkar, are you there?
Yeah, I'm here.
Okay, thank you. Any question from anybody?
May I ask one question, Vijay?
Yeah. Please, sir.
So it's an excellent work, Omkar. So I was just wondering, I know, more the N50, better would be the genome, but at some point of time, there must be, some kind of, holistic approach, to look into, say that, yes, this is the cutoff, for N50, especially, when you're going for such hybrid assemblies. So in that case, probably, what is suggested is, to go for L50 and probably U50, because the metrics would probably vary, and it would give you, a more precise assembly. So did you try looking into the other metrics?
Not in detail, but we have all the data, so we should look at that also.
Right. I mean, especially in this case, where you don't have a reference, especially N50. I know in N50 it is at some point of time, you say that more the N50, the better would be the genome assembly. But you're absolutely going in a more robust approach here, where you're going for a hybrid assembly. So in that case, probably it's always nice. It's always a good idea to go for L50 and U50. Yeah. So thank you so much. Thank you, sir.
Hello. Any more question? If no more question, we move to next speaker, Miss Kalpana. Kalpana, are you there?
Is Kalpana there? Is there? Is there?
Yes, sir. She is there.
She is there.
Kalpana, please start your presentation.
I can't hear. Is she there?
Yeah, I'm sorry, sir. I could see she's here. I don't know why. I think maybe she has some net problems, connection problems. Yeah. Kalpana, are you around, please? By the way, Kalpana is Professor P.K. Gupta's postdoc. Yeah. Sir is calling. Probably, we can just wait for a few minutes. We are just on time. Probably, should finish in 6-7 minutes.
Hello, Kalpana, are you there? Are you going to make a presentation? No, I can't. We can't see you. So make the presentation. No, but can you share the screen? But nobody can see you. You know, no, I can't see you. I can't see you. I can't see your screen. No, but the audience cannot see it. Prashant, she says that she's sharing the screen, but we can't see her.
Strange, sir. I mean, we could see her, and we could see her microphone on as well.
Now I can see her.
Yeah. Yes. Yeah.
Kalpana, I can see you now. Make the presentation. Share the screen. No, but the audience cannot see your screen. I can see you, but not your screen.
Kalpana, there's something called present now. At the extreme right. When you go down. We cannot hear you as well. There's something problem with your microphone. I presume. Would you mind looking into your slots whether or not the microphone is plugged into a good slot or not? Yeah. Now we could see your screen, but we cannot hear you.
So Kalpana, so can you hear us on the?
Yes, I am able to hear.
Everybody, now it's there. Come, start. Now we can see the screen presentation. Start.
So I'm going to present this work, Bioclues DNA methylation study for the research studies created by ALA 2883. So the main objective of this work was to understand the molecular mechanism. So
make it, Kalpana, make it full screen.
I have already did that, sir.
Okay, go ahead.
Academic resolution of the access fibers of downstream genes activated due to production that was created DNA code and its corresponding environment gene in pathogens.
No, Kalpana.
Hello?
No, the audience cannot hear you.
From this side, it is showing that better application window.
Prashant, can you hear her?
No, sir. I mean, it is not at all audible.
No, it is not audible.
Yeah. Sir, if it is not audible, maybe with your permission, you may end the session, sir. Maybe we can
Yes,
get to hear your recorded voice later. Yeah.
Kalpana, you can later on send the recorded presentation because we can't hear you. Okay. Thanks very much, Kalpana. Mm, no, it's okay. No problem. Okay, Prashant, we had a wonderful session, three oral presentations. Every presentation was excellent. It's, he invited talk. He invited
Yes,
talk.
sir.
One of was on regulation, and the second was Rajiv Bandyopadhyay, and the third was Harpreet. All three presentations were excellent. I enjoyed listening to them. Then there were three or four three oral presentations, and then yes, and I like to thank all the speakers who shared their work and wish them good luck in their future careers. And with this, we end this session. Thank you very much, Prashant, for giving me this opportunity of sharing this session.
No, we should thank you, sir, for your kind time and
And
energy.
thanks to
It's been,
Vijay, who
quite
conducted the
insightful
session very well.
having you.
Vijay, are you there?
Yes, sir. Yes, sir. I'll- yes, sir.
Vijay,
Yes, sir. Yes, sir. I am there, sir.
Vijay, can you hear?
Yes, sir. Yes, sir. I also thank Pers, sir, for uh, this giving this responsibility.
Okay, thanks,
Thank you.
thank you.
Okay, thank you, Professor P.K. Gupta, sir.
We conclude this session.
Thank you very much, sir, for the wonderful session, and there were very interesting talks. And thank you, Dr. Vijay, also. Be taking Professor P.K. Gupta's precious time at this age actually is a lot of inspiration for all of us. He is a source of energy for all of us, and definitely this session was really wonderful. And now I would like to call upon our next chairs. Those will be Dr. Sundarajan and Dr. Abhishek Kumar, because we are inviting our next keynote speaker, Professor Vinay Panda from JNU, and he will be talking on genomics and society relevance to data and common sense. And I already had an opportunity to listen to him in Central University Dharamsala year and a half back. I remember that. So I welcome Dr. Vinay Panda as well as our chairs for the next session. So Dr. Sundarajan and Dr. Abhishek, please over to you. Thank you very much.
Prashant, are we starting the session now?
Yes, please introduce the keynote speaker. Thank you.
Yeah. Hi everyone. We are going to have a wonderful keynote session now from Professor Bene Ranjan. He is currently a professor in School of Biotechnology, Jawaharlal Nehru University. But he started his career very long time. His research area includes data integration, genomic medicine, building, optimizing computational tools for high throughput data, and metabolic engineering of secondary metabolics and biodiversity, genomics and informatics. He was basically he did his MSc from Delhi and DPhil from Oxford University, and he lived in UK, USA, Japan before moving back in 2010 to India, New Delhi. And he also did got a lot of scripts and training in La Jolla, California, USA, and fellowship from American Cancer Society, and also a visiting researcher of genome science at the University of Tokyo. Vinay taught courses on microarray molecular diagnostics and at the University of California. He has been also director of Gannett Lab Foundation, Bangalore, and he got many awards. To name a few, it includes American Cancer Society postdoctoral fellowship, USA Felix Scholarship, University of Oxford Graduate Fellowship, and University of so many awards. He got a Radhakrishnan Memorial Beckwith Award. So today we are lucky to hear his talk. So we all welcome together, and let Professor Pandey start his lecture, the keynote address. It's on genomics and society, role of data and common sense. It's a very peculiar topic. I have seen a lot of Nature paper also on this topic. So we will be glad to hear from the professor. Welcome, professor.
Good afternoon, everyone. And I would like to thank both Prashant and Devendra for inviting me and giving this opportunity to talk to all of you about the work that we do and an exciting time that we all live in. The title of my talk is Genomics and Society, Role of Data and Common Sense. What I'm going to talk about today is how data has played a central role in our lives and how it all started, the birth of the field, what we call genomics today, and give you some examples where data has helped to understand biology better. And I'm going to give you 3 different examples from completely different fields, starting with the disease biology like cancer and understanding plant-based metabolites better, and then towards the end, talk about how genomics can help to understand very interesting species in our biodiversity and understand about the biology about those species even better. Towards the end, I'm going to talk about why understanding data and the way we produce and share data openly is extremely important to our future, and how common sense plays a very important role. With that, let me start and take you back to the history. How did it all start? And of course, this is not inclusive of all the great discoveries in science that has taken us so far and has brought us to this date. These are some of the landmark discoveries from all the way back 1870 onwards. I'm not going to read all this to you. As you can see on the screen, right from the beginning, in the year 1871, from the discovery of nuclein by Frederick Mascher, and following the chromosomal theory by Sutton and Boveri, it all started when scientists took interest in understanding the genetics, or what we call the hereditary basis of life, and followed many different important discoveries like discovery of all the 4+1, if you take uracil nucleotides by Albert Kosel, for which he was given a Nobel Prize. And then the most important thing, even if you don't believe it, the term genome, which was coined way back in 1920 by a German botanist, Hans Winkler. In fact, people didn't really understand what genome was then, but the term was coined, and he used the term genome multiple times in the paper that he published then. The most important and the golden period of genomics started in the second part of last century, when Erwin Chargaff came up with what we know as Chargaff's rule, both the first and the second parity rules, where he defined the nucleotide compositions in the DNA, and then the Hershey-Chase famous experiment where DNA for the first time was proven to be the genetic material, and of course the landmark discovery of Franklin, Wilkins, Watson, and Crick when they discovered the double helix structure of DNA, followed by Nirenberg and Khurana's discovery of triplet DNA codon. If you go to the present, in the last 50 years, there are many different important discoveries in the field of genomics. And again, I'm not going to read this. As you can see, the most important one are related to sequencing of DNA, starting from Sanger, Wally Gilbert, and Paul Berg, both the DNA sequencing and recombinant DNA technology in 1970s, all the way to the invention of the instruments that are known today are the next generation sequencing and anything in between. Now, what has happened in the last 10 years is nothing but a miracle in the field of genomics, because the amount of data that we produce has really gone leaps and bounds, which is presenting newer difficulties and challenges to biologists. And we'll talk about some of those in the next few slides. Genomics as a discipline actually started, or rather coined, the term genomics is coined in 1987 by a scientist who worked on, Tom Roderick, who worked in the Jackson Laboratory in Maine, which is the eastern coast of the US. And there actually started a completely new journal with the same name called Genomics. And in fact, if some of you are interested, there are very interesting anecdotal talks and newspaper articles about how this entire coining of the term genomics started in the local bar in close to the Jackson lab by Tom Roderick. The most important milestone, as all of you know, in the field of genomics started when there was an important effort to sequence human genome. And the project was coined then and came to be known as the Human Genome Project. The project traces its past to the Department of Energy in the US, and the effort was announced in 1986, but officially with the help of National Institutes of Health in 1990. As you all know, the draft genome was completed in 2000 with the landmark announcement in the White House by both the public and the private efforts, which was by the National Institute of Health as the public party and Celera Genomics as the private party. And the papers were published in the same time, both in Nature and Science in 2001, as the draft sequence and the complete sequence in 2003, which is exactly 50 years after the discovery of DNA as a double helix by Watson, Crick, Wilkins, and Franklin. Now, DNA sequences of human, what we know as the human genome sequence, is nothing short of a magnificent and unprecedented biological feat. Because can you imagine all that we work on today could have been possible without this sequence information? Hence, this is the resource, the magnificent resource which started many years back and we are using every single day. And therefore, towards the end, when I talk about data and why it is very important to produce data, good quality data, and deposit the data in the open domain so all of us can benefit, is an extremely important step. And this is just the newspaper cutting during the month of June 27, 2000, when the announcement came for the sequencing of the draft human genome for the first time. This is for the students in the audience, and this is a task for you to go back in history and identify these early pioneers. And all of them are pioneers in their own right, and every single individuals played a very important role in the field of genomics, and especially understanding human genome and doing the sequencing and analysis of the human genomes in early days. So it's a task for you, the younger audience, the younger crowd in the audience, to identify each of them and to know who they are and what their contributions are to the field of genomics. So I'm going to show you a few figures and take you through the last 25 years or so, how instrumentation or different innovation in the technology has helped us to sequence DNA better, faster, and cheaper. For example, this is a picture of 1994, how the early DNA sequencers looked like that helped us to sequence some of the genes during then. And then fast forward to 2006, these are some of the applied biosystems 3730, what we know, the automated DNA sequencer, which for the first time automated the whole process of sequencing, identifying the DNA sequences faster. And in fact, these are the instruments which are responsible for coming up with the draft human genome sequence in the year 2000 for the first time. And there are hundreds of them used both by the public as well as by the private effort. Now again, fast forwarding to 2020, as you can see, these are the Illumina HiSeq instruments, which helped for the first time to sequence the entire human genome of 6 gigabases or 3 gigabases haploid genome in just a matter of days. In fact, in less than 2 days. Again, if you fast forward today, we have the Illumina NovaSeq sequences. This is NovaSeq 6000, which helps to sequence multiple human genomes in the matter of hours. So as you can see, in the last 25 years, we not only have grown in technology, we have also grown how this technology has helped to produce genome data faster and cheaper and better, or with better quality. Now, this is a paper which is published in 2015 by Gene Robinson's lab. In fact, this is dated because this is, as you can see, 5 years old. But I wanted to tell you that this gives you an interesting idea about where we are going with the amount of data that has been produced by the field. This is a comparison of the data produced by the field of genomics with that from astrophysics and the common sites like Twitter, social networking sites like Twitter, and obviously very popular sites like YouTube. As the numbers say on your screen, the amount of data that is produced by the field genomics is already dwarfing those in other fields. Hence, the amount is a lot to deal with even today, and in going forward, we will have even complex problems to deal with the quantity of data. And towards the end, I'll tell you what are the solutions that the field or the scientists are thinking about to deal with this complexity of the data in addition to the amount of data. Now, this is a glimpse of where we are now in terms of the exact size of the data. There are multiple different databases today that exist that store human sequencing and non-human, for example, plants, bacteria, or virus sequences in the databases. And the largest one of them is the SRA, Sequence Read Archive database. As you can see on your screen, this data is, as of today, the 4th of December 2020, the amount of data, the total data, is nearing 46 petabytes. And it is growing very rapidly. Of course, this is from the one database. There are multiple different databases, and I have mentioned two other, the EMBL hosted database and also the Japanese databases, but this is by far the largest one. This gives you an idea about the humongous amount of data that is produced by the field genomics, including the data which is there in the database today. Now, as I had mentioned before, what are the drivers for this humongous amount of data generation? Now, access to instrument is one of the driver, because 25 years back, we did not have those instruments that can produce data cheaper, faster, and better. We do now. In addition to the access, the cost, the low cost of the production of data also helped in producing a large amount of data. This has almost democratized the field of production of data, and you can find this next generation of DNA sequencers in almost all big labs around the world, which has helped to push the data quantity upward. In addition to the access and the cost, the efficient capture, storage, and sharing and analysis and visualization tools has also helped to get this data faster. Last but not the least, when the data is produced faster, we need a computational tools, technologies to catch up with those that produce data. And in the last 2 decades, the price to performance ratio for compute and other IT infrastructure has also caught up. Therefore, we have actually advanced quite a lot, not just in the data production, but also data storage, access, and analysis. And these are all the parameters that are helped, or rather the drivers that are helped to push the data production and storage and analysis at a much faster rate. But the most important question that we are going to deal with in the next few minutes are, is it useful to have this data? We have produced the data in a large quantity, but can we understand biology better? And I'll give you 3 different examples. The first one is from cancer, and our lab works on head and neck cancers to be specific, and I'm going to give you one example. This is actually different anatomical sites of the tumors of head and neck. We specifically work on tumors of oral cavity and multiple different sites on oral cavity, but today I'm going to talk about the tumors or the cancer of the oral tongue to be specific. Now, these are some of the pictures, as you can see on your screen, is the histopathology of the tumor. On the top, I am showing you three different images. The left one is just a white patch. It's not a cancerous lesion yet, but some of those white patches turn and become cancer at a later stage. And at the bottom, if you can see, these are the different stages. First, from a complete hypoplastic site to the site which has now become cancer. And one of the important questions that we are asking with the help of data, can we understand which one of those patches turn into tumors? And if we do that, it will be much better to manage these patients with tumors better. We can identify the patients with the tumors before even the white patches become tumor. And I'll show you one particular question that we have asked and try to answer with the help of data. Now, as you will see in a minute or so, this has turned into quite an interdisciplinary science, because we want to understand this cancer at the level of the genome, at the level of the clinical manifestation of the representation, and also taking both the data together by doing or by utilizing the computational tools to understand and answer the questions that we start with. Therefore, we have a specific clinical question in mind, and we use different tools and technology encompassing all these different fields which is there on your screen to ask that particular clinical question and try to answer if you can do that with the help of data. Therefore, integration of data is very important, and integration of multiple different types of data and different dimensionalities in those data, and ask the question, can it help to answer the question that we start with? And as you will see in the next minute or so, we have been successful by using data from multiple different sources. First of all, producing data, high throughput data on these tumors, and collecting clinical data from those tumors and patients, and integrating those data with the help of machine learning tools to answer a very specific clinical question. And towards the end, this is an important point I'll reiterate. It is very, very important to start with a very specific question in mind and go back to answer that question with the help of data, if you are a bioinformatician or a computational biologist. Now, these are the papers that we have published, which essentially precedes before we ask the integration of the data. Well, first of all, we have to produce the data. So these are the papers which are cataloging different molecular determinants, starting with single nucleotide variants, mutations, different germline and somatic mutations, copy number variations, gene expression changes, methylation changes in these tumors. Now, once these data are produced, we actually took this data along with the clinical data and the epidemiological data to ask a very specific question. And in this case, we asked a question, can data help in identifying metastatic and recurrent tumors a priori? Now, what have we done? We have taken data on three different levels. Molecular data, as it suggests in the left-hand side column, with different types of molecular data, starting with somatic mutation, all the way to copy number variations and the context of that data in the genome. Then the clinical level data, the HPV or human papillomavirus infection in this data, the grade of the tumor, the stage, the node, and the habits, for example, whether they have tobacco positive or tobacco negative, and whether the tumor has actually recurred or not. And the third, the final data that we have taken is something which is already published on head and neck tumors, but not from our geography. For example, the cancer genome data, which is already deposited, the cosmic data, the International Cancer Genome Consortium data, and data from individual laboratories which have elucidated functional roles of some of those molecular determinants. Once we have these data together, we try to integrate them under a machine learning platform and a framework that I'm going to describe next. we call this machine learning platform or the framework as Cafe Mocha, which is an integrated tool to analyze different type of molecular determinants to find clinically relevant signature. This is agnostic to the tumor type, although we have used this here to understand head and neck tumors. You can use it for any different tumor as long as you have the data and a specific clinical question in mind. This is a paper that we published in 2018 in Journal of Clinical Oncology, CCI, and if you are interested, you can go and read the paper. But very quickly, I'm going to show what exactly this paper will talk about. These are the different type of events and the molecular determinants we have taken. Some of those are categorical events like somatic mutation, some of those are quantitative events like gene expression, and some of them are functional events. For example, copy number changes where it is linked to its gene expression. As we know, if a gene copy is higher, we perhaps expect the gene to be expressed at a higher level, and if it is, we have taken that into consideration. And similarly, methylation changes linked with up or down gene expression changes which are linked to that. And once we have those data, we have devised a new, what we call as complete specificity margin-based clustering framework to understand that the relevance of a molecular determinant to ask a question related to the metastasis and recurrence of the tumor. In this case, I am showing in this figure 3 different stages. For example, whether it is a metastatic disease or a recurrent disease, or there is no metastatic or recurrent disease. And by each of the dot on your left-hand side is a single molecular event, where in this case, expression and methylation, which are linked. And once I know that which one of those events are linked with a specific clinical determinant, I can actually use this particular clustering method. And I'm, due to time, not going to define or not going to go into the details. Some of you who are interested can go and read the paper. And once we know that which particular molecular determinants are specific to this event, we only select those. And therefore, we maintain a very high specificity. This is the final signature that we have obtained, which we call as MI44, which are basically 44 different molecular determinants for this particular type of cancer. And as you see at the end, we have validated that with an independent set of tumors, which gets a very high sensitivity. And because of the framework that we use, the clustering method that we use, we wanted to retain a very high specificity. And of course, as we get into more data, we will have different sensitivities in different type of data. So this is a beginning effort to understand whether we can integrate data to ask and answer a specific clinical question, and we have achieved that. Now, most important question that we want to answer, does integration matter? That means, is it important to integrate multiple different types of molecular determinants? And I'm going to show you in the next figure that yes, but please keep in mind, the importance of integrating multiple molecular determinants depends on the clinical question that you ask. In this case, I am obviously showing you from the 1 to 6. There are 6 different events that, as you saw previously, we are taking into consideration. And the height of the bar actually talks about the number of total molecular determinants which are responsible to give us the sensitivity, and the little black dot here talks about the actual sensitivity. Hence, in the last one, where we have a single combination, obviously when we take all the 6 different types, we get the highest sensitivity. Although if you reduce that to number 5, that means instead of taking 6 events, if I take 5 events, there will be 6 different combinations. And here it's not that bad, although it's not the highest, but we still get close to 75% of sensitivity. And obviously it goes down as I reduce the number of events. This gives you a glimpse that why integrating different molecular determinants is important. And as I said, it depends on the clinical question that you ask. Okay, so far, hopefully I have demonstrated that data helps to understand human diseases like cancer better, to actually answer a specific clinical question. Now, let's move on to the second example. The second example is from a completely different field, because the intent of the present talk is to let you know that data can help in biology. And in the second example, we are talking about a plant-based metabolite. Here we are talking about the plant which is the neem plant. Almost all from India know about this plant because this plant is a very utilitarian plant. Through history, we have used different part of the plant for multiple different purposes. And for today's talk, what is important is that let me go back one slide here. The important metabolites which are a part of the fruit of the plants, and there are more than 130 different secondary metabolites which are present in these fruits. And in today's discussion, I am only going to focus on a single metabolite, which is called azadirachtin, because this is an important, both agriculturally and pharmaceutically important metabolite, which is commercially exploited as well. Now, before we answer the question, can data help? We are the first group in the world to elucidate the genome and the transcriptome with a series of papers published. As you can see here on the screen, we did sequencing analysis and interpretation of genome and the transcriptome of this plant, and with a very important intent that we want to understand these pathways of this secondary metabolite biosynthesis better. Now, as I told you that the kernel of the seed of this particular fruit, neem fruit, is very important because it stores the secondary metabolite. Now, I'm not going to go through these particular slides. Basically, there are different classes of terpenoids, and the terpenoid that today we are talking about is azadirachtin, which is in the fruit. Then we started with a very specific question. And what is the question? The question is, can we use artificial intelligence, and in this case specifically deep learning methods, to identify fruits and leaves with high metabolite content? And why is it important? It is important because the metabolite content in fruits varies between fruits and among different fruits of different region. And what we wanted to understand and work with is, can we use image-based deep learning methods to identify fruits and leaves which have very high metabolite content? In other words, can I just walk into a tree, take a picture of the fruit, and can say which fruit has higher amount of metabolite or not? Now, this was a serendipitous thought in our mind when we started it a few years back, and to our utter surprise, this method actually worked. Now, in order to do this project, we have traveled length and breadth of our country. So this is the orangish, figure that you see is basically the part that we have traveled to collect the neem fruits and leaves, which we have taken the images. And the blue southern states that we have done is that we have collected the fruits and leaves independently as a validation set. So this is again, published in this preprint bioarchive, and we are in the process of modifying it, getting it peer-reviewed for the publication. So what is the premise here? The premise is that we have taken more than 80,000 images of fruits and leaves with the intent that the fruit images can actually be a surrogate to understand the intracellular concentration of azadirachtin. So we have two different inputs here. We have taken the image of the fruit and the leaf, and we also have biochemically, experimentally determined the metabolite content by using HPLC. So we have a HPLC derived actual concentration of the metabolite, then we have an image. So these are the part of the training set. And once we have that, we also have augmented the images, which has gone the part of the deep learning training set. Once we have done that, we ask the question, can we identify, or do the images represent what is there inside the fruit? And again, here I'm not interested to know the exact amount of azadirachtin, but I'm interested to know high, mid, and low. These are arbitrary, like cutoffs we have put up. So for example, I'm interested in anything which is more than X, I call it a high content. Anything between Y and X, mid. Anything below Y and Z, maybe low content. So that once I know whether it's a high content or low content, I can only select those trees for collection. And this is the result. To our utter surprise, when we used more than 83,000 fruits and 86,000 leaf images, we get a very high accuracy in terms of validation. As you can see, the 70% accuracy. Remember, this is the first time we have used this to use any images of the plant parts to understand the intracellular content class of this particular metabolite. As we speak now, we are refining this model further with more images and with more calculations, more computation to understand and to push this accuracy even further. Okay, so based on that, we have also made the desktop and the mobile app, And the idea here is that you can walk into a tree with your mobile phone, take a picture of the fruit, and instantly it will tell you whether the fruit has high or low content of the metabolite. And therefore, if you are interested in this metabolite, you can only choose those which has high content of metabolite. Therefore, it actually saves you a lot of energy, time, and effort not to go to the trees which has lower amount of metabolite. Okay, now hopefully I have demonstrated the use of data, a very different case in the plant. Now let's move to the final example. Again, a very, very different example. This is a nascent example, very early stage. We have actually just begun working on this, although we have demonstrated 3 years back the proof of concept. Now here, the use of genomics data to understand snake venom beta. The snake in question here is called Russell's viper. This is one of the venomous snake, beautiful animals, venomous snakes in our country. And there are more than, believe me or not, more than 1 million snake bite related deaths in India in the last 2 decades. There's a paper actually published in 2011 plus on neglected diseases, which talks about there are more than 60 different venomous snakes in India, but most snake bites, most if not all, are due to 4 different snakes: spectacled cobra, saw-scaled viper, Russell's viper, and common krait. And Russell's viper, the picture that you see here, is one of them. And the questions that we wanted to ask is very difficult to get permission to catch these snakes, because snakes belong to restrictive species. So it's actually prohibited to catch those snakes just for fun or even for research, because you need permission. So we wanted to ask the question, can we use the shed skin? Because snakes shed the skin periodically. Can we use this shed skin and get reliable amount, quality, and quantity of nucleic acid which can be used for sequencing? And once we use those for sequencing, can we actually do or get an acceptable quality of assembled genome? And finally, if we do that, can genome sequence be useful to understand the snake toxins? And I'll show you the proof of concept data for this paper, which we published actually in 2017. The answer to the first 3 questions is yes, and we don't know the last one, because we're still at a very early stage of understanding snake toxins, because it's a highly complicated cocktail of chemicals. Now, this is the paper that we published in 2017, which we have finally could use the shed skin to obtain reliable and high quality DNA and sequence the DNA, assemble the genome to get an acceptable level. As I'm using the term acceptable because the quality of the genome still could be much better by using multiple different technologies and by using multiple different tools to assemble those. But the quality is still acceptable to understand the toxins of this particular snake. Now, here I will just take a break, and so far, I hope I have demonstrated the use of data in 3 different, completely different fields, but equally well how genomics and genomic data can be useful to understand biology better. Now, where does the field take us further? Now, let's talk about a little bit on the data. What do we need to do about the data? Now, most important thing about data is that it has to be de-linked from the investigator. For example, if I am producing the data, you should not be, as a researcher, you should not be getting back to me and asking the question, can you give me the data? The data must be deposited in an open repository so that everybody can get it. There should be a common set of rules and the procedures, how to generate the data, how to share the data, and how to deposit the data. The data QC metrics has to be standardized, and the data, which goes without saying, must be shared ethically, must be shared equitably, and must take care into account if it's a human data, privacy, and we must have law and regulation that actually govern how, who, and when to share the data. Now, in addition to what I just talked about data, as you can see here, generation, access, sharing, security, findability, interoperability, and reusability of the data is very important. Therefore, we need to have a national policy. I'm very proud to say that I'm one of the few people who actually drafted the India's first national biological data sharing policy, which is pending cabinet approval. And once it's approved, it will actually become a national policy which will govern how we share data with other researchers openly and in a responsible fashion. Of course, it goes hand in hand with the establishment of the National Biological Data Center, which is in progress, so that all the data which is produced can be actually housed in a common place and can be shared openly, equitably, and in a responsible way with all the researchers. And I'm not going to read this particular slide, but as you can see, we need to be putting our efforts how data is produced, which is there on the left-hand column, and what are the different expertise that we need, which is in the middle column, and what are the infrastructure that we need to ask questions. The important point here I want to mention that the bioinformatics expertise, because I'm expecting most of the audience in this talk are going to be from the field of bioinformatics and computational biology and genomics, is very important to distinguish that bioinformatics expertise not necessarily always equals to the expertise to deal with data in a responsible way. Therefore, we need to have training modules to train our future generation of scientists in this field, not just how to generate the data, but how to deal with the data in a responsible way. And I'm going to show you few solutions that I have in mind which can help doing that. Now comes to the common sense, and this is the part which is very important, particularly for the young researchers who are thinking of establishing their own labs in the field of bioinformatics, computational biology, and genomics. Now, here is an important perception, and I'm sure that all of you face this some or the other time. And this perception, dare I say, is by most biologists, if not all. They think that the people who work with computers don't do any work. It's the computers who do the work. The real science is actually done by a biologist, and bioinformaticians are just a helping hand. They are just helping them to get from place A to place B, which is absolutely not true, because computational biologists or bioinformaticians actually do help more than help. They can generate their own hypothesis, and they can answer the biological question in a much more meaningful way by using data, as I showed you in the 3 different examples in my talk. Now, I have a very funny video to demonstrate what most biologists actually think, and I'm going to play that video to you. So here you see the guy in the black t-shirt is the biologist, and the train is the biological science. And the biologist is thinking that he's the one who is actually stopping the train, but actually what is happening, the train engine which is stopping the train, not him. And like stopping, it is again him who is going to think that he's the one who is going to start the train to go to the next level. Now he's trying to push the train. Little does he know that actually it's the engine which is pushing the train. This is of course a joke, but you get the point. Okay, now what is the solution? What can be the solution? All of you who is listening to this talk, is very important to be open. Do not ever be closed with your data. Put all your data in open domain. And you must always make sure before you try to publish anything, put your publication, the article, in a preprint server. You don't have anything to lose by doing that. You have everything to gain. Don't be afraid to work on tools only. Remember, tools are an essential part which will make sure that the discoveries are correct. Because if you work with the wrong tools, all your discoveries may turn out to be wrong. Work, if possible, with a question or a bunch of questions in mind. And as you'll see in my next slide, stay away from the metrics. And one of the metrics which is overused, not just in our country, but all over the world, is the use of journal impact factor. And you must stay away from this. I'm sorry for the haziness of the slide, because it's probably just image which I've used from the publication. And here I'm showing you x-axis the retraction index, and the y-axis is the impact factor. So more the journal impact factor, more is the retraction index, which means that the chance of getting the article or getting an article retracted is much higher if the journal impact factor is high. In fact, There are innumerable number of examples why the journal impact factor is one of the bogus, unscientific tools you can ever use, or metrics you can ever use to judge the quality of science in a particular paper. If you happen to be in the committee who is deciding, do read the paper yourself. That's the best way to find out whether the investigator has done good work or not. It may not be possible, I understand, in all cases due to time, but certainly not by using the journal impact factor as a surrogate. And similarly, the H index, stay away from this. They do not define who you are. That doesn't define the quality of science. What is rather going to define the quality of your science is the paper that you publish, how good it is, whether you have actually answered or tried to answer important biological question or have developed an important tool. Now, these are the last slides that I have. I can only tell you that the lessons that I have learned in the last 20 years working myself, you only work if you are passionate. That means you must be passionate in the subject that you're working with. Work with the questions in mind. Don't belittle the tool, as I mentioned before, because tools are very important. Give importance to quality over quantity. Okay, and not everything requires complex computing and doesn't require complex algorithm and large data. Sometimes small amount of data is also good enough to answer the question. Keep that in mind. Avoid jargons. Try to talk in simple language so that everybody can understand. Be humble. This is the most important point I want to tell you, especially the audience who are young people, students, young scientists who want to establish their own lab. Be humble. That's very, very important. Avoid focusing on where you publish and hence avoid the journal names, but focus on the quality. And as I mentioned before, avoid journal impact factors and H index. Enjoy science and the path of getting to the answers. Final slide, I would like to acknowledge the people who work in the lab, the hardworking students, the project fellows, the postdocs, funding agencies, collaborators in and out of the country, and many distractions. As you can see, I love Star Wars, so these are the two great characters that I like, both Yoda and Anakin Skywalker. Finally, the wise words, I'm sure you'll agree, of George Bernard Shaw, who told, the reasonable man adapts himself to the world, the unreasonable one persists in trying to adapt the world to himself. Therefore, all progress depends on the unreasonable man. So my request is, don't be shy to be that unreasonable man. Thank you very much. And with that, I would actually love to take your questions.
Wow, it's amazing. Yeah. Thank you, Professor Panda. It was an amazing talk. We enjoyed, and the depth and breadth, how things are going. Yeah. Any questions?
So I'm audible?
Yeah.
Yeah, I am audible.
Yeah.
So this is not directly a question. I need to congratulate the speakers because I really appreciate. And this is a time where a lot of people are sleeping after having heavy lunch. So I got excited by this particular talk and his special example, and especially some of the quotation he's highlighted, and very much important. So I need to ask you one thing. Using this leaf data for the quality analysis of understanding of secondary metabolite, how many variables they can be used, and what are the basis? Audible or not? So when you use the second example of the leaf deep learning algorithm, where you want to from image data to know the percentage of secondary metabolite per this particular plant, the leaf data. So I need to find out how many variables you have taken for the construction of machine learning models, or the deep learning example. Now you understand it? I'm not getting any response. I'm audible or not?
Yeah, we can hear it. Yeah,
Yeah, I'm not getting any response. That's why I'm asking it. Is clear or
yeah,
not?
it's audible. Yeah, we can hear
No,
you.
actually,
Uh,
Sundar Dada,
okay, okay.
I think Vinay Panda's microphone is muted. Maybe he's away. I don't know. Vinay, would you
No
mind
issue, no
unmuting your
issue. The
microphone?
question.
He's
Sorry,
responding in
can you
the
can you hear
yeah.
me now?
Yeah, I can hear you now. Yeah, please.
So I don't really understand the question, but if I can guess what you're
No,
asking,
I'm now. Can you hear me now? I'll be
no,
repeating the
your
question
voice is
again.
very choppy, so I cannot hear you properly.
Okay, okay, no problem. No. Yeah, go ahead. What you understand? Let
If I
please.
understood what you want to ask, both the
Yeah.
images of leaves and fruits totaling about 86,000 each were taken along with the biochemically derived metabolite to make the training set. So the variable that goes into that is both around, we can say, 170,000 images along with the biochemically derived value of the metabolite.
So I need to ask you which features you have taken for construction of machine learning models.
The
The
features
number
are
of
not being
features.
extracted as separate, but it is a CNN model, so it doesn't actually extract a feature.
Okay.
It takes the
Okay.
whole image into
Okay.
account.
So there is no option of selecting the features which can be used for the construction of models. They automatically selecting all features by automatically.
Yes, there is nothing in CNN
Okay.
that you can figure out extraction of a particular feature. It takes the image as a whole.
Okay. So this is a what do you say, deep learning model?
Yes.
Okay. Okay. So can it be same method can be used for the other type of data set? Or because I have some plant based data set which I need to identify that these are wild plants which is grown widely and they have medicinal potential. So
It
can these
depends.
Veta need? Yeah. Okay.
It depends.
Okay.
You cannot just say that this is going to be universally applicable. It may or it may not.
Okay. Thank you. Thank you very much. Take it.
Yeah, if I may ask, with the permission of the chair,
Yeah.
Sundar and Abhishek, yeah. Thank you so much, Binay, for your wonderful talk. I just typed in the chat box. I'm not sure if you have seen that. So I was just wondering, in India, I've had a privilege to work with Professor Kavikishore, who is also the next keynote speaker. He works on, something called, dental cancer. But at this point of time, we don't know. Although we know okay, that certain desired phenotypes like HNSCC or dental cancer or oral cancer, and although we know by the names, but we don't have at least some common set or common minimum set of genes that are probably responsible for such diseased phenotypes. So can we at some point of time take a conglomerate of all the next generation sequencing based approaches like the MedDeep-seq data sets, RNA-seq data sets, the ClipSeq or HideSeq seq or something like that, and come up with a common most differential express genes that are probably responsible for the diseased phenotypes, just like what we have for the housekeeping genes. So I know efforts have really been made into this direction, but not towards understanding the diseased phenotype. So your valuable insights would be helpful. Thank you so much.
So I still don't understand what you mean by common sets of genes and diseased phenotype. If you think the disease in terms of a germline mutation, then the answer is no, because most of the diseases that I talked about are predominantly somatic. Therefore, the changes that accompany these diseases are mostly known. Now, if you are talking about expression changes, that of course it is going to be helpful to understand which genes are expressed high or which genes are expressed low in a particular disease. But remember, the disease is a somatic disease. It is an disease which is induced due to consumption of certain substances like alcohol or smoking or chewing tobacco, or for example induced by the presence or absence of an infection. Therefore, it is going to be specific to the population in which it is there in response to a habit, in response to an external stimuli. So expression data, I showed you some of the expression data. It is there, but of course more the better. If we have more data linked with some of the genetic variations, or for example epigenetic variation, it doesn't hurt. And having a consortium will obviously help if we can put together with a specific phenotype. But I must also warn you that the tendency of the bioinformatics community tends to be that we have more data, it's actually going to be better. It's not necessarily the case. What is more important is the quality of the data. And if you are working on clinical samples, what is more important is you need to have a very good set of well annotated clinical samples. This is going to define your success or failure, not necessarily the amount of data.
Yeah, thank you very much. In fact, indeed, I was talking only about the somatic, diseases, somatic mutations only. Yeah, thank you. Yeah.
Any other questions from the floor?
Hello.
Yeah.
Yeah, I have a question, sir.
Yes, Madhumita.
So in case of leaf and fruit model, the question was that can deep learning help to identify fruits or leaves with high metabolite content? It means the model is designed by taking some of the parameters of the image captured to like predict what's the metabolite content. So I just wanted to know what are the features that is fed to the model. Features, when I say feature, I mean the like what are the feature of the image that has been captured. Like I want to know.
I think I just answered that question. You probably were not listening.
No, I'm listening, but I'm confused, sir. It's like,
so you do one thing, you go and read the paper. It's all there. If you have any questions, send me an email.
Okay.
Madhumita, you just answered it. The CNN takes care of everything. Once you feed the total number of images and things. So we don't need to fiddle around the what parameter or anything.
Sir, actually, I was having a further question. If CNN takes everything by itself, so it might be dependent on the quality of image we are feeding. So we have an app, so the app can capture images in different ways, right? So will that model be affected on the camera quality of the app? That's why I was asking what are the features they are going to feed in CNN.
No, usually the picture should be as clean as possible, right? It's obvious, even for human eyes. See, we have to see how the human eyes treat things. If we cannot see an image clearly, we cannot understand. Same thing goes to the machine model. If machine model cannot recognize things in a clearer way, it cannot do anything further, right? So it is obvious we should know things should be as good as possible so that things can move on.
Yes, sir. Thank you, sir.
Okay. Any other question? Okay, Binay, it was a very good lecture. But I want to ask you one thing. We should have when we should stop. That's the main question I used to always think. We add more data from complex and whatever way we get. But we should have an understanding when we stop adding further data to our model.
So it depends again on what type of model you're using. Usually in the deep learning model, more data is going to validate it better. And the number actually depends on an imputation. So some people have come up with some model which suggests X amount of data you already saturate. It depends on the model. Each model is different and each model will have its own saturation point.
Yeah, thank you. Okay, thanks for the wonderful lecture. We are really honored, and it was very good to walk through various edges on this line, how things are happening, and what are the good point in thinking the ethical, all these things. Thank you so much, Binay. Prashant, you want to add anything? Yeah, we can
And
have
I'm
a
not
group.
sure.
Yeah, we can have a group photo with Binay. We can switch on the video.
I'm sorry, my camera doesn't work.
Ah, my goodness.
Thank you all. See you later.
Thank you.
So thank you so much, Sundarda and Abhishek, for this for sharing this wonderful session. It's been very insightful. It was a wonderful talk from Binay Panda. So now I just take pleasure in inviting Arvinpreet Kaur and Sakshi Singh. Arvinpreet Kaur has been our student co-chair for this INBIX conference, and Sakshi Singh is a woman in biology co-chair. So they'll just take us around through something called elevator pitch, where Nancy and Sakshi they will announce our names. We just need to introduce ourselves in less than 30 seconds. So the best answers now could probably might win a good award. Yeah. Thank you so much. Over to you, Nancy and Sakshi.
Thank you, Parash. Am I audible?
Yes, you are audible.
Yeah. Hello, welcome you all. First of all, I'm going to introduce myself. I'm Sakshi Singh, DST Inspire Fellow, pursuing PhD in Bioinformatics from Banaras Hindu University. Well, my research area is in immunoinformatics, and I have worked on data analysis like RNA-seq data analysis as well as microarray data analysis. Along with, I have worked on protein modeling as well as MD simulation methods. So apart from this, I'm also working for a Women in Biology Forum, which is a part of Bioclues. This is a small introduction of mine, and now I'm passing to Arvinpreet Kaur. Arvinpreet, over to you.
Yes, good evening, everyone. This is Arjun Preet Kaur from HLB College, Jalandhar. I'm a master's student. I'm pursuing MSc in Bioinformatics. So, I would like all of you to introduce yourself for 30 seconds. Firstly, I request Dr. Abhishek Kumar to please introduce himself.
Hello, is it you asking me
Yes,
to introduce myself?
yes,
Yes,
sir.
sir.
Okay, so I'm Abhishek working at the Institute of Bioinformatics, and our group is focusing on computational genome biology and various aspects of genomics, primarily cancer genomics and neurological disorders, plus marine uh, omics. We try to tap marine resources, says microbes from marine resources, and try to evaluate drug-like compounds. And we have several training working with us during this year. We have 10 students who have done primarily their master thesis, and uh, about two who did two no four of them did their BTech degree. So we are very extensively believing that if I want the person who is trained, I must train. And so with this, we are working, um, doing research and training.
Okay, thank you, sir. So, everyone will introduce themselves for 30 seconds. Also, if any of the mentors want a project, a student for project, can also mention this. So next one is Anshita Prasad. Please introduce yourself. Hello, Anshita Prasad. Can you please introduce yourself? Okay then. Anaswara Ramachandran. Please introduce yourself. Next one is Arunima De. Please introduce yourself. Arunima De. Please introduce yourself. Dr. Ashok Kumar. Please introduce yourself.
Hello,
Yes,
so I'm
sir.
audible.
Yes, sir.
So yeah, मेरा नाम अशोक कुमार है. I belong to Punjab University and मैं 17 साल से कैंसर बायोलॉजी में काम कर रहा हूँ. And we published few papers on bioinformatics. पर मेरा basically area cancer biologist हूँ मैं. After PhD completion of PhD, I started working on this bioinformatics field. In this starting, when we complete with the two PhDs, I was not interested which area I will be taking. So we take a area of drug repurposing, and we published this particular paper in 2018. And we, after 2 years, it is published in a scientific report, and we are just got top 11. It is ranked top 11 in the all ranking by Nature Communication. I am very excited about it. The work we have started is a serendipity is well recognized by throughout the world. Now I'm excited to teach student also. I'm mentoring for more than 4 students for the PhD. I'm married, happily married, and I have two twins kid. That's all about me. Thank you very much.
Thank you, sir. Next is Ayam Gupta. Please introduce yourself.
Hi. Good evening, one and all. So my name is Ayam Gupta. I am working as a PhD fellow in the Birla Institute of Scientific Research. So my main objective for this project is dissecting the vitamin K pathways. We are doing that in the using next generation sequencing data. Uh, I have been working with Dr. Prashant for more than 1 year now. Uh, that's it. Thank you.
Thank you. Next is Dr. Devendra Biswal. Can you please introduce yourself? Okay. Then Diptin Laskar. Can you please introduce yourself? Next is Disha. Can you please introduce yourself? Pawan KM. Can you please introduce yourself? Himani Sharma. Can you please introduce yourself? Hridyoy Jyoti Mahanta. Can you please introduce yourself?
Hi, good evening. This is Dr. Hridayjyoti Mahanta. I am involved with the Advanced Computation and Data Science Division at the CSI and NIST. My division is headed by Dr. G.N. Sastry, who is also president of this Bioclues. And I have done my PhD in applied cryptography. And after joining here, I am trying to work on the artificial intelligence and machine learning, and trying to incorporate those applications towards drug discovery. Thank you.
Next is Dr. Jairam Valadi, sir. Please introduce yourself.
Hello. You want me to ask questions?
No, sir. Everyone is giving introduction. It's a elevator pitch round.
No, you want me to. See, I just joined, so that's why I'm not able to get it clearly. Yeah. Yeah.
So
Hello.
everyone is introducing themselves for 30 seconds.
Yeah.
Elevation pitch round is going on. So please introduce yourself for 30 seconds.
Hello.
Yeah, Jayaraman sir and P.K. Gupta sir, I'm sorry, this is something called elevator pitch, which we have introduced. So each one of you are requested to introduce yourselves in less than 30 seconds, please. Thank you so much.
You want me to introduce myself? Yeah.
Yeah, I mean to the audience, less than 30 seconds, although everyone knows you.
I just got in a minute.
Thank you so much, sir. Thank you.
Yeah. So I am Jayaraman. Currently, I am working at Flame University in the Computer Science Department. I am retired from NCL Pune about 10 years back, and after that, I had been working at CDAC, and currently I am visiting in Shivnada University and also Pune University. Currently, I teach machine learning. I have expertise in applications of machine learning in bioinformatics, chemoinformatics, chemical engineering, and I also work on process control and further applications. But my current interest is applications of machine learning and bioinformatics. Thanks a lot.
Thank you, sir.
Yeah.
Next is Krishna Mohan. Can you please introduce yourself?
Yeah, thank you. I'm Krishna Mohan. I work at Birla Institute of Scientific Research. Currently, I have about 3 projects. One is on the diabetes of the Indian population, where we are looking at exploiting the endogamous groups, that is like Agarwals from Jaipur and Reddys from Nellore and Chettiars from Chennai. So we have done the deep sequencing and the genotyping of very large families, that is including 2 or 3 generations, and also first and second cousins, where in which families the diabetic incidence of the diabetes is very, very high. That is almost like an average is around 15 to 16% in the world population, whereas these families have more than 30% of their members has diabetes. So this way we have identified around 8 loci which have not been reported previously, which we are validating right now. And I have other projects which are on the cardiovascular diseases and also on the oral cancer. Both of them are correct. Thank you.
Thank you, sir. Next is M. Sawood. Please introduce yourself. Next is Madhumita. Please introduce yourself. Next one is Malik Yusuf. Please introduce
Hello.
yourself.
Hello.
Okay,
Am I
Madhumita,
audible?
please
Yeah.
introduce.
Yeah. Good evening, everyone. This is Madhumita. I am a senior research fellow at the Department of Bioscience and Bioengineering at IIT Jodhpur. I am doing my PhD under the supervision of Dr. Shushmita Paul. My research interests include computational biology and bioinformatics, pattern recognition, soft computing. At present, I'm working on a project where I'm trying to design some algorithms based on pattern recognition that can be helpful in integrating multiomics data to identify cancer subtypes. Thank you.
Thank you. Next is Malik Yusuf. Can you please introduce yourself? Then Meera Indrakanti. Can you please introduce yourself? Nagamani. Can you please introduce yourself?
Hi everyone, myself Dr. Silvaraman Nagamani. I am working as a scientist in CSIR Northeast Institute of Science and Technology. So I am mainly associated with our director, and mainly I was completed my postdoc in this group. So since my postdoc, mainly I work on the developing open source drug discovery software, which is known as molecular property diagnostic suite. Thank you.
Thank you, sir. Next is Nandini Dey. Please introduce yourself. Nasreen Bano. Please introduce yourself. Next is Niharika RD. Please introduce yourself. Nimi Freeman. Can you please introduce yourself?
Hi, I'm Nimisha. I'm currently pursuing my PhD from Central University of Kerala, and I'm working on drug transcriptomics. And I did my MSc from Cochin University of Science and Technology from Ernakulam. And currently I'm working with Dr. M. Nagarajan. He is my PhD guide. Thank you.
Next is Om Prakash. Can you please introduce yourself? Next is okay, Om
Hello.
Prakash. Yes, yes, sir.
Hello, sir. Can you share my slide?
Om Prakash, this is not the time, for your presentation. Your talk is tomorrow. It's not today, please. Please understand.
Okay. Thank you. Thank you.
This is something called elevator page. हम आपको ये रिक्वेस्ट कर रहे हैं कि आप 30 सेकंड में आप खुद आप इंट्रोड्यूस कर सकते हो आप. ठीक है? Thank you so much.
Is it okay if I go ahead? Because my name would not be displayed here. We are logging from a single computer.
Yes, please.
Okay, thank you. So, good evening, everyone. I'm Nidhi Shukla. I'm currently working as a DST Women Scientist Fellow at Birla Institute of Scientific Research with Dr. Prashant. I've been here for almost 1.5 years. I'm using next generation sequencing approach to study mutational signatures in prostate cancer in Indian cohort. And the main focus is on long non-coding RNA as our lab is expertise in that. So this is all I have. Thank you.
Thank you, Nidhi.
I have like 2 more colleagues, so they'll also introduce themselves.
Sure.
Hello everyone, myself Shalini. I'm a PhD student working on dissecting vitamin K pathways in humans using next generation sequencing analysis under the guidance of Dr. Prashant. Thank you.
Hello everyone, my name is Sonal Gupta. I have completed my PhD from
BISR Institute under Dr. Krishnamohan and Dr. Prashant. In my PhD, I have worked on the role of mutation studies in a rare anorectal malformation, that is congenital pouch colon. Currently, I am working as a research associate at SMS Medical College, Jaipur, and there I am working on a project titled as where I'm looking on a metabolomic profiling in mother's milk across the Rajasthan. Thank you.
Thank you, Sonal. Thank you, Sonal.
Next is Dr. Prashant. Please introduce yourself.
Yeah, thank you so much. I'm Prash. I'm currently at Birla Institute of Scientific Research. I've had about 7 years of postdoctoral experience after having finished my PhD in 2009. I'm currently working on dissecting vitamin K pathways in human transcriptomes, funded by and large by DST. I also have been recently funded a prostate cancer project, for which we have been extensively working. I think my group has already been have introduced themselves, and without them, this extensive research would not have been possible. And with the wonderful team spirit and leadership of Dr. Krishnamohan. Thank you so much.
Thank you, Parash.
Next is Dr. Next is Rajul Jain. Please introduce yourself.
Hi, my name is Rajul Jain, and I am JRF in the DST sub project and a research scholar in the Albag Educational Institute. My research focuses on to see the effect of various selected pesticides on human ortholog neurodegenerative diseases using model organism C. elegans. And here in Bioclues, I am a mentee under Sundar sir and Renuka ma'am, and we are working to build a deep learning based model to see various drug target interactions. Thank you.
Thank you. Next is Sylvia Vescuse. Please introduce yourself. Dr. Sylvia, please introduce yourself. Then Sundarajan, sir, please introduce yourself. Hello. Dr. Sundarajan, sir, please introduce yourself. Am I audible, sir?
Yes, very much, Nancy. Maybe Sundar, might be meditating now. It's his meditation time. I know him because he's my mentor. Yeah. I think next you may go ahead, please. No. Yeah. By the way, Sylvia was here. Sylvia has been one of the foremost Bioclues members since 2007, I believe. She's an eminent scientist from Peru. She is 68-year-old scientist, and she's doing her another master's. Although she's a PhD, she said that she wanted to do a master's in data science, and she's doing that. I think the young audience over here can take some inspiration from her. Thank you so much.
Hi Prashant, anything you're asking me?
Are Dada, I was just thinking about it. I just thought you're meditating. So if you can quickly introduce yourselves in 30 seconds, please. I know we know you very well, but please.
Yeah. I started my career as a mathematician, did my mathematical honors. Then suddenly, sometime back, somebody was telling that the computer science has come in the 80s and they wanted to learn. But I was not very keen. But one of my friend came and say he paid all the money, but he cannot understand anything because he's very weak in mathematics. He cannot understand algorithm, all what the computer does. Then he pushed me and forced me to join the computer science on behalf of what he paid already. Then I started loving the computer. Then I realized we are able to command a computer, which is a slave, and get it done anything. So that was a challenge I started, and over 20 plus years, I was enjoying this. And then also I start how the data science is growing very well, and the data is coming. And the parallelly, we see the softwares and platforms are changing over, and it makes us to be more active and learning and things. It's a big cycle of business and also knowledge, and also how the ability of handling more and more and more data is possible. And that's why we had a beautiful session from Binoy also, how and when we should stop. We add more data, it doesn't going to be useful or not useful. It's a very big question. We have to answer all this. So the data science I enjoyed last 20 years plus, and I have done a lot of modeling and things. After coming to Singapore, I joined the NUS for PhD in computer science. We did a lot of algorithm and handling many bioinformatics data initially. Then I went for a banking industry, and did a lot of customer modeling to choose a customer for a particular plan. And then I went to NTU to see how we can select students course material, because in a bigger classes like 500 students, the teacher cannot do very well in pay special attention to student, but can it be automated? So all these kind of things were able to answer through data science. And also now the clinically we have a lot of things, how the medicine is going to be helpful, and what are the way we can get it help in but it may take a while for the medical science. It is still we are getting data, and it's not completely okay to get the things modeled up, but eventually it will happen. All things are going to happen. Only we have to wait and see how things are going to happen. Even like the personalized medicine, all this is not an imagination, but it's going to be happening maybe after a decade or so. And also we require a powerful computing system, platforms, people who want to make it work. These are the things. So I'm enjoying all the things with the data science, and I welcome people who
Greetings,
are
everyone.
really
My name is
interested to have some projects from me. But I'm going to be grilling like people. If you don't answer me quickly in few days, then I will just say thank you, bye bye. Because we need people to plan. Don't plan in a daily scale. Plan on a 7-day scale, so that you can moderate and you can achieve what you want to do within a week. That's the main thing. Don't plan wrongly. Like today I will do 10 things. You can never finish it, because you don't know which one will longer time or shorter time and things like that. So always plan on a weekly scale. Then always I do like a weekly scale planning, and so far I am very successful. So that's my advice to all the team members also I say. And we can win always, because there will be a lot of problems. No power cut, there will be a power cut, or there will be a water problem, no bus, no electricity. These are things, but still you can see people like Abdul Kalam. He has no light in his home, but he made light in everybody's life in India. He became a role model. So these are the lot of examples we have from India. But outside, of course, we don't have a shortage of anything. Everything is surplus here. We never have a power cut or shortage of water, or we have a clean water, clean pollution, no pollution here. Still, but we are able to do good things, because we don't need to bother all this, so we are able to do better. That's why outside people are able to bring out more things, because they are more thinking on what they are able to do, not what they are going to eat or what they are going to do or things like that. Basic amenities are automatically available to them. So that's my advice. And any question from anyone? Hello, any question?
I think no one has question.
Sir,
Okay.
What?
yeah.
What,
I think
Arvind?
Ashok sir is having a question.
What, Arvind, you're telling?
I guess someone is having a question. I don't know who is having.
I think still we have 34 participants.
So I guess, sir, no one is having any question. Thank you so much
Okay,
for
no
your
problem.
inspiring
Thank you.
words.
Welcome. They can, if they feel shy in the open platform, right? They can email also. That's the main thing. Your shy should not end your curiosity. That's the thing. You don't kill your curiosity by shy. It's okay. You cannot be meeting the audience, but still you can write back to us. We will be answering you as soon as possible. Okay.
Thank you so much, sir.
Okay. Welcome.
Next is Sunil Kumar Polipalli. Can you please introduce yourself?
Hello.
Yes, sir. Please introduce.
Hi, this is Sunil. Hi. Hello.
Yes, sir. You're audible. Please introduce yourself.
I am Dr. Sunil. I am working as a cytogenetist in Loknaik Hospital, New Delhi. I am looking after the genetic disorders regarding to pediatric population and looking after the prenatal diagnostics in thalassemia, mutational screening part, and looking after the LSDs, lysosomal storage disorders also, and the sequencing part also we are doing little bit, but we are not into completely into bioinformatics, but we are still in the learning phase only. Thank you.
Thank you, sir. Next is Vineet Upadhyay. Please introduce yourself.
Hello, my name is Vineet Upadhyay. I joined Institute of Bioinformatics for my PhD under Dr. Abhishek Kumar's guidance. Our main area of research is genomics of human diseases and disorders, especially cancer genomics. I like to experiment and learn new things in bioinformatics and really appreciate the amount of data information which is present in open domain so that anyone curious can learn readily because my background was in biochemistry and during my dissertation project I grew interest in bioinformatics and thus I'm pursuing it for my PhD. Thank you so much.
Thank you, sir. Next is Vineet C. Please introduce yourself.
So, assuming that everyone has done this elevator pitch, I think let's not waste our time. I think let's thank Sakshi and Nancy for their wonderful job. Thank you so much, Nancy and Sakshi, and thank you everyone. Yeah. So, without wasting time, we are just dot on time. It's 5:47. So, I would like to invite Ayam Gupta and Santosh Kumari to take care of the video abstracts. We have had about 16, I believe. Now that was my count, but Ayam would take us around to the various video abstracts. These video abstracts, what are these video abstracts? Ayam will be able to tell us. So Ayam has already introduced himself. Santosh Kumari, unfortunately, she is not here today. She is our another joint secretary. Yeah, thank you so much. Over to you, Ayam. Yeah, I'm just stopping the screen.
Yeah, good evening, one and all. So now we are going to start our video abstracts. So the first video abstract that we are going to present is predicting long non-coding RNA protein interactions in prostate cancer using combination of computational and biophysical methods. So the authors are online. So after playing the videos, they'll take the question answers. We set the limit to 3 minutes. And 1 minute for question and answers. So with that, I'll just start the video abstracts. I'll start sharing my screen now. I'll share this. I hope my screen is visible. Okay, so the first video abstract is presented by Nidhi Shukla. Here it goes.
Greetings everyone. My name is Nidhi Shukla. I am working as a Women Scientist Fellow at Birla Institute of Scientific Research, Jaipur. Along with my colleague Ayam Gupta, we are presenting a poster titled Predicting Long Non-Coding RNA and Protein Interactions in Prostate Cancer using a combination of computational and biophysical methods. So before moving ahead, I'd like to give a brief background so that everyone stays on the same page. Prostate gland is a part of male reproductive system, and as you can see from the left picture, that it lies behind bladder around urethra. Whenever there is any abnormality, it eventually leads to prostate cancer. It is one of the leading causes of cancer-related deaths. And if I talk about India, it is one of the most prevalent cancers. And as you can see from the figure in the right side, that it has spread to almost all parts of the country. If I talk about the statistics, according to GloboCan 2018, there are almost 26,000 prostate cancer cases, out of which 17,000 cases have resulted into deaths. So as far as we know, there are not many next generation sequencing studies on PC in Indian population, and as genetic variation is best seen in the non-coding RNAs, and as our lab has expertise in long non-coding RNA, we aim to identify candidate mutations associated with it. We also want to decipher the regulatory role of identified long non-coding RNA using long non-coding RNA protein interaction studies.
To achieve this, we did a pilot study on prostate cancer samples using whole exome sequencing approach, for which we ran the in-house developed and benchmarked whole exome pipeline. Post the VCF generation, we did downstream analysis using VT, applying various stringent filtering criteria. More detail is provided in our published paper. This study resulted in identification of many variants in the samples. The number of variants between cases and controls with respect to depth greater equal 5 and lower equal 20 respectively is shown in the Venn diagram on the right hand side. After finding out the variants, we prioritized 30 causal genes and blasted them against the non-code database, which is the database for long non-coding RNAs. Doing this resulted in 12 hits, out of which 5 were again shortlisted based on their expression in the prostate cancer and interactions with other prostate cancer causal proteins. At the end, 3 best hits were selected. Using the computational biology approach, we further tested the shortlisted LNC RNA for LNC protein interaction using LNC Pro Server, as this method has a good ability to discriminate interacting, non-interacting RNA protein pairs and can also predict the RNA protein interactions within a given complex quite reliably.
So far, we have identified 3 long non-coding RNA which can play an important role in the tumorigenesis of prostate cancer. So the LinkPro results need to be further validated using biophysical approaches such as microscale thermofluorescence and pull-down assays. We are also trying to identify the regulatory elements of these long non-coding RNAs by genome walking, where we are trying to identify some enhancers as well as CTCFs, and together all these will provide us possible leads towards biomarkers and development of novel therapies for prostate cancer. Thank you everyone for your kind attention, and we would be happy to take any questions. Thank you.
Yeah, okay. So this was presented by Nidhi Shukla and myself. So any questions, if there are any questions, we would be happy to take those. I I think there are no questions. Even if there are questions, please do send drop them through email. You try to reply to each one of those. With that in mind, let's look at the next uh, video presentation. I'm just sharing my screen again.
Hello everyone.
So this presentation is from Sonal Gupta. She is presenting her work on molecular interaction studies to predict the mutational genes for understanding long non-coding protein regulation. Let's start this. The author is online again. If there are any questions, please do ask the authors.
Everyone, today I
Thank
will
you.
be presenting on the topic entitled Molecular Interaction Studies to Predict the Mutational Genes for Understanding LNC RNA Protein Regulation. This is a small overview on a rare anorectal malformation, that is congenital pouch colon, where the colon ends in a pouch-like dilated structure which is communicating with the genitourinary system through a large fistular opening. This is
I
a
think
small
there is some issue
background
with
for the
the
present
audio.
study from the
Let me
whole
just
exome
troubleshoot
sequencing
that.
of
Please
16
give me a
probands
minute.
of
I'm so
total
sorry for the
64
inconvenience.
samples. Variants were called through a benchmarked in-house pipeline in our lab. From this, the causal variants in the form of misRNA protein interaction pairs using docking complexes. Firstly, the LNC-RNA non-heterocyte SAT 2007. Hello everyone. Titled Molecular Interaction Studies to Predict the Mutational Genes for Understanding LNC RNA Protein Regulation. This is a small overview on a rare anorectal malformation, that is congenital pouch colon, where the colon ends in a pouch-like dilated structure which is communicating with the genitourinary system through a large fistular opening. This is the small background for the present study from the whole exome sequencing of 16 probands of total 64 samples. Variants were called through a benchmarked in-house pipeline in our lab. From this, the causal variants in the form of missense, frameshift, stop gain and stop loss mutations were found. Furthermore, from whole exome sequencing, we have also identified a LNC RNA
Uh, Dr. Prashant, I would request you to play the video from your end. I think I'm not getting any audio.
Yeah. 1 minute. Yeah.
Can you please help me with this regard?
Yeah. Sure. I'm doing that. That was what I was just trying to do. That 1 minute.
Hello everyone. Today I will be presenting on the topic entitled Molecular Interaction Studies to Predict the Mutational Genes for Understanding LNC RNA Protein Regulation. This is a small overview on a rare anorectal malformation, that is congenital pouch colon, where the colon ends in a pouch-like dilated structure which is communicating with the genitourinary system through a large fistular opening. This is the small background for the present study from the whole exome sequencing of 16 probands of total 64 samples. Variants were called through a benchmarked in-house pipeline in our lab. From this, the causal variants in the form of missense, frameshift, stop gain and stop loss mutations were found. Furthermore, from whole exome sequencing, we have also identified a LNC RNA which was showing interaction with one of the causal gene, that is KIP13A protein. This is the small workflow for deciphering mutational signatures in LNC RNA protein interaction pairs using docking complexes. Firstly, the LNC RNA non-HETSAT 002007 was predicted from CPC whole exome sequencing, which has shown a putative role in regulation. We have selected KIP13A protein as one of the CPC causal gene from our erstwhile study published in 2018, which was showing interaction with LNC RNA. Then the probable RNA binding sites in KIP13A was predicted using preprint server. The KIP13A was modeled with each fragment of LNC RNA using HDOC web server. Then the interaction probability between RNA and protein was done using RPA-PRED server. Finally, the effect of mutation on protein nucleic acid interactions were done using support vector machine tools. This is the figure showing RNA interacting residues in KIP13A using preprint server. This is the figure. Second is the figure showing secondary structure of LNC RNA as predicted by RNA fold version 2. To investigate the interaction of LNC RNA with KIP13A protein, we have first divided the long non-coding RNA into 7 fragments according to the predicted secondary structure of LNC RNA. The 3D structure of each LNC RNA fragment were docked with kinesin motor domain of KIP13A using HDOC web server. The highest affinity was predicted with G fragment of LNC RNA with R1 region of KIP13A. Then the R1G docked complexes were further refined with HDOC server. To further validate the docked model, positive and negative control studies were carried out. Finally, the effect of mutation on protein nucleic acid interaction was studied using MCSM-NA tool. These are the conclusions that have been inferred from the present study. The mutational studies carried out showed distinct effects on protein stability and binding affinity to the nucleic acid. This study has led us to map the binding regions as putative candidates for identifying mutations in LNC RNAs, which were found causal for CPC and make us to understand their role in disease regulation, which could be further targeted for specific treatment therapies. I would like to acknowledge these
Hello
people
everybody,
for
my name is
helping me
Madhusmita Raut and I'm going
in
to present
conducting the
today
present work. I would
the
like to
therapeutic
thank
topic
Team
entitled
INVIX for giving me an
System Biology
opportunity to
Analysis to
present my
Identify
work
Key
here.
Molecules
Thank
Involved
you.
in Gest.
Excellent talk. So the Sonal is here and she'll be happy to take any questions. I think there are no questions. I would like to ask one question from the author. So the KIF13A is is it a quite well characterized protein, or does I'm not very sure about this. Can she shed some light on this?
Yeah. Hello everyone. Yeah, as we know that long non-coding RNAs, they are a very long base pair fragments. So to perform docking between LNC RNA and protein, we have to first divide this LNC RNA into various fragments. Then we have modeled each fragment of LNC RNA with a different model complex with the KIF13A, and when we achieve the best docking score and the best model complex, that we have taken further for our validation studies. Yeah, thank you.
So thank you, Sonal. So I think we should move on to if there are no questions, we should move on to our next video. Dr. Prasad, can you play the next video?
Senior Professor and I'm Dr. Praveen Mathur, Senior Professor and Head, Department of Pediatric Surgery, SMS Medical, for a rare called as congenital pouch colon, in which the large bowel exhibit enormous dilatation. An unusual feature of CPC is the prevalence of this malformation in the Indian subcontinent, with the highest in north and west part of the India. The exact embryogenesis and etiology of this anomaly is not known. In order to understand the molecular basis of this rare disease, we have performed whole exome and transcriptome sequencing on 64 samples. We have identified candidate mutations and interesting long non-coding RNA, which we believe could be considered as biomarker. This protocol would be the first step towards achieving this for any disease, be it rare, complex, or Mendelian. Thank you.
Another excellent video from our previous segment. This was the elevator pitch done by Dr. Praveen Mathur. I think he's not here as of now. So, with that, our next. So I would like to introduce Avanpreet Kaur. She would be.
Today, I'll be presenting a video abstract on the title Comparative Proteomic Analysis of Pseudomonas pseudoalkali genes, Protein Organophosphorus Hydrolysis 2, OPHC2 in Mesophilic, Cytophilic, and Halophilic bacteria. Over the last 7 decades, we have been using pesticides for the better crop production, and over 47% of these pesticides contain phosphoric acid, which are very much toxic to human beings as it stops the neurotransmission in the body, and over 200,000 deaths are reported annually due to organophosphate poisoning. So we need to remove them from the environment. Earlier, we used to adopt physicochemical based remediant technologies, which were very costly, time consuming, and causes secondary pollution in the environment. So we need to develop bioremediant technologies, and with the advancement of biotechnology and recombinant DNA technology, which we can do it in a much better way. And we found a very promising candidate, OPHC2, that is organophosphorus hydrolase 2, in a mesophilic bacteria, Pseudomonas pseudoalkali genes. So we need to know its structure in a much in order to know its functions and apply it for bioremediation. So we did a small project to know its amino acid composition, which will pave a way to find its structure. So we took the faster sequence of organophosphorus hydrolase 2 in Pseudomonas pseudoalkali genes and found its homologous sequences using BLAST-P, and we made a list of different mesophilic, cytophilic, and halophilic bacteria using BacDive database. And after that, um, we used XPC prot param tool in order to know the amino acid composition of different mesophilic, cytophilic, and halophilic bacteria. And we took the average and standard deviations of mesophilic, cytophilic, and halophilic bacteria, and then we draw a graph in order to get the deviation and the amino acid composition and compare mesophilic with cytophilic and mesophilic with halophilic bacteria. Um, this would help to make a genetically modified bacteria for the extreme environments, because mesophiles, like we need to develop such bacteria from the extreme environmental conditions. So we need to know its amino acid composition, and after that we can know its structure in a much better way, with which we can develop a genetically modified bacteria and apply it for bioremediation. Thank you so much.
Yeah, I am,
Excellent
with your
talk,
permission, I
Avanpreet.
just wanted to
Are there
make
any
a
questions?
small comment. Yeah, I just wanted to make a small comment.
Yeah.
Yeah. So Nancy, we have what do you call Abhishek here, who can really help in your projects in this particular area. He's already here. He's watching all these video abstracts since he wanted, some insights into this direction. Yeah. Thank you.
So if there are any more questions for Avanpreet, she would be happy to take.
It's
I
also
think she is
here.
online already.
Hello.
Hello, everybody. My name is Madhusmita Raut, and I'm going to present today.
Hello. Am I audible?
Yeah. Please, yeah.
Okay. So my mentor, Dr. Ashutosh Kumar, is also here. He guided me in this
Oh,
particular
wonderful. Yeah.
project.
Yeah. Yeah, wonderful. Very nice. It's absolutely decent work. Yeah, congratulations. Yeah.
Yeah, thank you, Arvind.
And thank you.
With
Again,
that,
my
we
name
would
is
move
Madhu.
on to our next presentation.
Topic entitled System Biology Analysis to Identify Key Molecules Involved in Gestational Diabetes Mellitus. A part of my PhD work, which I did in Vellore Institute of Technology, Vellore. At the outset, GDM is characterized due to glucose intolerance caused by insulin resistance during pregnancy, mostly in the third trimester. Around 15.8% of the population affected by hyperglycemia in pregnancy, out of which 90% of the cases are gestational diabetes mellitus. GDM is caused due to a lot of genetic predisposition, demographic factors, and environmental factors are responsible, as well as there are a lot of epigenetic modifications such as DNA methylation, microRNAs that play a role in causing GDM. In order to understand that, we proposed a research concept of identifying genes involved in GDM and through system biology approaches, understand the crosstalks between GDM affected mother and placenta. The workflow proposed was to identify the genes involved in GDM through literature and then segregate them to maternal and placental, and thus understand the regulatory functions, comorbidities, associate cell types, as well as through system biology approach, construct a protein-protein interaction network, understand highly interconnected genes and transcription factor regulatory network, as well as microRNA regulatory network to understand the regulation of these factors in affecting these genes, thus understanding the role of these hub genes. We constructed interactome of protein-protein interaction network, maternal and placental, the regulatory function network, the associated comorbidities, as well as the cell types in which these genes were expressed. And then we wanted to know the highly interconnected genes, for which we used three regulatory factors such as degree betweenness, closeness, and we identified 5 hub genes for maternal and 3 for placental. And when we map these hub genes on pathways, the maternal genes regulate in insulin secretion and signaling pathways, as the placental genes responsible in amino acid transport pathway, as well as the beta cell dysfunction pathway. These genes were intertwined and had interconnections among each other to regulate various maternal and placental pathways in GDM affected mothers. Thank you, and I would like to take any more questions.
So it's a post abstract.
Hello
So
everyone.
Madhusmita
We
is
are
on.
working on dissecting vitamin K pathways in humans using next generation sequencing analysis. Vitamin K is a fat soluble vitamin that affects coagulation pathways within the body. It is essential for the synthesis of coagulation proteins. Natural vitamin K includes phyloquinone derived from plants and minoquinone derived from intestinal bacteria and fermented foods. Vitamin K obtained from the diet is considered to reach the target tissues via lipid absorption and the transport system. Once transferred to the cells of the target tissue, vitamin K is metabolized by redox cycling in the intracellular endoplasmic reticulum body in a process known as vitamin K cycle. Warfarin is an oral anticoagulant drug, inhibits VCAR, stops the vitamin K cycle, and prevents the gamma glutamyl carboxylated conversion of the blood coagulation factors, thus inhibiting coagulation. Activity of both GGCX and VCAR are regulated by calmolin. Recent evidence has revealed that GGCX is the only enzyme involved in GLA formation based on structure and function analysis of GGCX at the gene level and animal studies showing that GGCX gene deficiency causes embryonic lethality from systemic bleeding. The objective of a study is to transcriptome study of healthy and unhealthy samples from adults and infants and study of SNPs to understand their reactions and novel transcripts which related to vitamin K deficiency, and validation of SNPs, which will used as biomarkers.
There are a number of clotting factors present in blood plasma which require vitamin K for activation. At present, 17 types of vitamin K proteins are reported to date. Coagulation factors 2, 6, 9, and 10, and anticoagulation proteins C, S, and Z all depend on vitamin K1 in liver synthesis. Osteocalcin, MGP, GAS6, GRP in the tissue outside the liver rely in vitamin K for post-transcriptional modification. However, the function of PRGP1, PRGP2, TMG3, and TMG4 remain unclear. Vitamin K deficiency is not very common in adults and is usually associated with specific conditions such as malabsorption disorders, antibiotics, and drug interactions, especially with coumarin-based anticoagulants or an extremely poor vitamin K content diet. All neonates have reduced vitamin K at birth, and vitamin K deficiency can contribute to various comorbidities such as significant bleeding, poor bone development, osteoporosis, and increased cardiovascular disease. In addition, a high intake of vitamin K decreases coronary heart disease and the risk of cardiovascular diseases. Our future work is on identifying the differentially expressed genes and SNPs in the vitamin K pathways. We would like to gratefully acknowledge all of our team members. Thank you very much.
You mentioned
Shalini is online. If anybody would like to ask some questions.
Morning all. Myself
Uh,
Anusharya Ramachandran. I am a integrated master student with coronavirus. Morning all. Myself
okay,
Anusharya
if there are no questions, maybe Santosh Kumari's video is remaining, Dr.
Upon it's yeah.
I'll try to share from my end if the screen share, if the
As a scientist and
okay,
reference
if it's working for you.
scholar working under guidance of Dr. Sugna Karbure sir from Lovely Professional University, and my subject area is microbiology, and I'm working on cervical cancer, HPV, and today's abstract title is Low and High Risk HPV Phylogenetic Analysis and in silico protein interaction studies and docking studies in cervical cancer. Cervical cancer is the second largest cancer among women in India, and according to WHO, every 8 minutes a woman dies of cervical cancer. It is caused by persistent infections of high risk and low risk HPV types. It also causes other cancers like oral cancers, throat cancers, and head and neck cancers, and vaginal cancers, anal cancers. So there are three vaccines like Cervarix, which is bivalent, and Gardasil is quadrivalent, and Gardasil 9 is nanovalent, which are used for those types of 9 types of HPV and 4 types of HPV and 2 types of HPV. And Gardasil is available in India, and Gardasil 9 is not available in India. And there are more than 200 types of HPVs among animal and human species. So the main objective of my study is to construct a phylogenetic tree based on E6 and E7 oncoproteins of high risk and low risk HPV types, to construct a protein interaction studies and docking studies based on that. So the methods and materials we used here are the PAVE database, which is used for papillomavirus. So that data is retrieved from PAVE database, and the phylogenetic tree is constructed by using MegaX software. And protein interactions studies have been done with string software, and protein docking studies with Hex software. hex software and PDB format files. PDB file format is being taken here. So the results and we got, and we are working on it, like phylogenetic analysis shows evolutionary relationship of a high risk and low risk for better understanding of the cervical cancer, in which we got the 21, 26, 51, 73, and 82 HPV types, which show anal cancers, vulvar cancers, vaginal cancers of in low risk in those cancers, and that transform as a high risk in cervical cancer. So they show a single branch, and they are closely related in the phylogenetic tree. So the protein-protein interaction studies, also the proteins which we have taken, like EGFR and VGF and GRP78 and laminin 5, ATM. All this shows that EGFR shows shows interaction studies with all these. And last, the protein blocking studies, where L1 ligand studies, ligands or L1 capsid proteins of HPV, and with other receptors, it shows good binding affinity with EGFR till now, and that is followed by heparin sulfate, which shows binding scoring and also structural affinity, or also very good. It is the prediction is in good in these two, other than the GRP78, laminin, and immunoglobulins interferons when we compare with this. And further studies has to be conducted, and we are just working on it. And thank you.
So that was the presentation from Santoshi Kumari. The next one is
My project is in-depth studies and experimental work, super nano fertilizers. This project is guided by Sudhanshu Singh from Amity University, Rajasthan. Let's go to the introduction. Our study is to develop control release nanofertilizers for promoting development of environmental friendly and sustainable agriculture. Our study applied nanoscaled and nanostructured materials for the production of super nano fertilizers. We used biofertilizers like rhizobium, azospirillum, azotobacter, and pseudomonas in combination with neem-based silver nanoparticles. Then these are tested on selected crop varieties of Fabaceae family, ensuring slow and efficient release into the plant system. They are developed in such a way that they retain properties like high solubility, stability, effectiveness, and less ecotoxicity. The methodology we used are we use some beneficial organisms to prepare biofertilizers, namely rhizobium, azotobacter, azospirillum, and pseudomonas. Firstly, these bacteria are isolated by respective methods from soil samples by using serial dilution techniques. The second part of the work is generating silver nanoparticles from neem leaves extract. We use 3 mm silver nitrate in 100 ml distilled water, then added 5 ml of 3 mm silver nitrate solution to the rest. Various techniques such as DLS and UV visible spectrophotometer quantized the silver nanoparticles we produced. The results are the required microorganisms for the preparation of respective biofertilizers are cultured successfully. Then we used charcoal as carrier to combine all these beneficial biofertilizers into a single mixture. Next, synthesis of silver nanoparticles by biological method is done, and this liquid formulation is added to the powdered form of biofertilizer,
and to make it into a complete nano biofertilizer. We tested the above prepared nano fertilizers on rice crop varieties and potato plants, and their controlled release on root and shoot growth was observed. To conclude, the major advantage of the above fertilizers are they yielded high they have yielded high crop varieties, providing good nutrition to the consumers. Due to its slow release property, there is reduced environmental damage. We develop them in such a way that they retain maximum beneficial properties. They are tested for their efficiency by physical properties like root and shoot growth and translocation of nutrients, etc. It is hence positively proved that they have effect on improving the solubility of nutrients, increasing bioavailability, and hence finally nutrient uptake efficiency. I want to acknowledge my guide Sudhanshu Singh for his guidance throughout the project. And I want to thank Dr. C. Manjulata, Principal Government City College, for her valuable support and allowing us to use the infrastructure of the college for forwarding our research work. And I also want to thank Mrs. P. Pushpalata, head and faculty, for extending her support for validation of nanoparticles at the prestigious IICT Hyderabad. I want to thank Bioclues.org and INBIX 2020 Conference Committee for giving me this wonderful opportunity to present my paper. Thank you.
Excellent abstract from Rani Durga. I think she's online, so if there are any questions, anybody can shoot. There is a question: use of this biofertilizer is cost.
Integrated masters.
Okay, so we'll take those questions in the last 5 minutes. I think we should continue on with the videos first. Please do post all the questions in that chat box. We'll go through them at the end of the session. Thank you.
I am maybe, if she wants to answer, that will be nice because there are only just two. So maybe if you want to take up the questions first. For nanoparticles.
Yeah. So Rani, is Rani? I don't see Rani online. That's why I was saying that.
Oh, is it? Okay. Yeah. It's okay. Yeah.
Hello, sir.
Yeah, she's here. Yeah.
Okay.
Niharika, what I am saying is you have a question in the chat box. Would you like to answer that?
Yes, sir. Sure.
Yeah. So the question is that the use of this fertilizer is is it cost effective?
Yeah, sir, presently, as we used silver nitrate, I can't say it is cost effective for time being, but we are trying to cut down the cost at the part in the part of generating nanoparticles, so that then I can say my work is cost effective, sir.
Okay. Thank you very much, Rani. I think that satisfies the question.
Myself, Anuswara Ramachandran. I am an integrated master's student in Central University of Tamil Nadu. And I have recently done a paper on soft matter physics and its relation with coronavirus. We all know that the world is going through a pandemic, COVID-19, which all started in November. And it's been a 1 year through this pandemic. We have all been going through a lot like quarantine, lockdown, etc. And the world has been on a break. And this paper mainly focuses on how the soft matter physics and its properties are related with the coronavirus. And I have discussed what all the forces are acting in coronavirus, like attractive forces, repulsive forces, van der Waals forces, interactive forces, etc. all have been discussed in here. And I have calculated the infection probability and how coronavirus is transmitted in people and how its probability is increasing. And I have found that it's been increasing like an exponential graph, rapidly increasing. We can find out through this. And I have discussed how to prevent it using an SMS method. SMS is not a phone mechanism. I am telling about S for sanitizer, M for mask, and the another S for social distancing. And how these three fields relate with soft matter is also discussed in my paper. And I have also conducted how to develop vaccine through soft matter physics is also being discussed in a brief introduction of my paper. And that's all. Thank you.
Um, yeah, that was the video abstract from Anasura Ramachandran. Are there any questions for her? I think there are no questions, so we can move on to the next candidate, Praharshit Sharma.
2020 delegates. This is Praharshit Sharma under supervision of British Kumar Raj at Indian Institute of Information Technology, Allahabad, Prayagraj, Uttar Pradesh, India. Our topic for the video abstract for Index 2020 is trigonometric compositional analysis of DNA sequences using binomial expansion theorem with coefficient perturbations. So, what is the rationale or motivation behind the work? Basically, we are assuming that for the sake of simplicity, a particular piece of DNA, cDNA sequence consists of purely non-low complexity, unambiguous species, namely adenine, thymine, guanine, and cytosine. And now it is evident that when we do an n-gram analysis of such a DNA or cDNA sequence, it's always normalized in the sense that the relative frequency of so purely from a compositional data analysis perspective, we can begin with a 0-gram analysis. So as to say, 4 power 0 is 1, right? So with 0-gram analysis, we consider that the 80 percentage in DC percent composition or computation, and fit the composition analysis of further n grams into natural number exponentiation of the trigonometric identity, which we know as the famous Archimedes or Pythagoras identity. In terms of trigonometrical function, sin square x plus cos square x is equal to 1. So the objectives of this work are to potentially characterize any DNA or DNA sequence using singular value x as per the fundamental trigonometric identity equivalence to Pythagoras theorem. So that is the KTGC. 80% is 0.5, GC percentage is 0.5. So 0.5 plus 0.5 is equal to 1, and sin square x is equal to cos square x is 1 by 2, and x is 1 by 4 in the acute angle regime. And acquiring this equation, we have sin square of 4x plus cos of 4x plus half sin square of 2x equal to 1. And so as well, we get x is equal to pi by 4 radians, or in sexagesimal system, it's 45 degrees. And result reduction further expanding to 2 grams, we get sin square x plus cos square x power 4 is sin square x over sin square x square cos sin square x into cos square plus 4 sin square x cos square x cube uh, and cos square x power 4. Yes. So in this way, we can see that for any number of n mers or k mers, we get the exact number of terms which can be equated with coefficient perturbations of this binomial expansion. For 64, which is going to be triplet frequency, right? So we can cube this equation, we'll get 64 terms. And it may be noted here that subject to Taylor series expansion, sin x and cos x can be approximated to any desired position purely in terms of x as per the two infinite sequence below. In conclusion, the sequence working scheme based on compositional data analysis perspective, purely using fundamental trigonometric identity and its binomial expansion is presented here. Working all subparameters in the coefficients of the Pascal's triangle of the triangle X value as described above. From such a deterministic approach, then we say that trigonometric genome compression can be achieved and perfected as a genome sequence in question. For example, H19 Homo sapiens 3.3 gigabase per can be retrieved using X axis, which can explain GC percentage bias in the quadratic case, because if you see the quadratic case, it's going sin per 4x plus cos per 4x plus half of sin squared 2x is equal to 1. Yes, no. Okay, thank you. To transform to not transform, a theme paper on compositional data analysis from the Center for Ecological Science IC Bank for reference here. Yes. Thank you very much.
Excellent presentation from Prahar Sharma. He has given us a lot to think about. I'm I'm not very good with mathematical stuff, but still, he explained it in a very Is I think Praharshit is not here for taking any questions, but if there are any, we would email him and ask not just him, but all of the authors. If there are any questions, we would just take them. Uh, so, okay, I think there are no more questions, and this concludes the day. This was our last segment. I would hand over to Dr. Prashant for the.
Yeah. Yeah, thank you so much, Am, for wonderfully repertorying and sharing this session. So it's been very insightful. But I think there are many missing faces, from the elevator pitch as well. Usually, the elevator pitch has been absolutely very successful on site over the last two occasions. There were many people, who were really enthusiastic, to come forward and discuss openly, their problems, so that the mentors over here, can try to solve you. So if there are no things, I think tomorrow we should probably meet at 9:15 exactly. Please join 15 minutes well in advance. You're going to have the third keynote tomorrow. And I think let's thank all the speakers, all the session chairs, and all the wonderful audience over here, and most importantly the conference chairs, Devendra Biswar and Utkarsh Raj. I think Dev is here, so Dev, would you like to give any final comments before we say goodbye? Yeah, for today. Yeah. So I think Dave might be busy, but tomorrow we have wonderful keynote that will be followed by a great machine learning session. We got very enticing talks, so please don't give it a miss. These are very important, and that is also followed by an industrial this thing and a panel discussion on bioinformatics in Northeast, where revered scientist Dr. Madan Mohan, who is DBT advisor, ex-advisor, is coming to the panel discussion. He was there here in Jaipur as well in 2017. So I think this has been a wonderful INBIX 2020 start today. So thank you so much, one and all. Thank you, Ayam. Thank you, one and all, and thank you, all of you. So we are meeting at the same link tomorrow. Please join at 9:15 a.m. IST. Thank you so much.
Thank you,
Bye-bye.
Prashant. Thank you all speakers and keynote speakers. Thank you very much. It was a wonderful thing today. Enjoyed a lot today. Yeah. See you all tomorrow.
Yeah. Thank you, Dav. Thank you. Bye-bye.
Bye-bye.
Bye, all of you. Bye, all of you.
Thank you, everyone. Thank you, Dr. Prashant, for organizing such a great event. It's been great that we have been part of INBIX. I will join right on time tomorrow. Thank you.
Yeah, thank you. Yeah, it's been a team effort. Thank you so much, all of you. Bye.